A very happy morning to one and all present here. It gives me colossal delight in welcoming each one of you to NEUGLIA 2021 Day 2. Yesterday, the symposium started with a welcome note by Dr. Sujata, Associate Professor and Head, Department of Biochemistry, Dean of Academics, Self-Supporting, Ethraj College for Women, which was followed by the association report by Dr. Malati, Associate Professor, Department of Biochemistry, Ethraj College for Women. It was followed by the special remarks given by a convener, Dr. Ilangovan, Professor and Head, Department of Biochemistry, University of Madras. The inaugural address was delivered by Dr. Prakash Babu, Secretary General, SNCI. Dr. Saravana Babu and Dr. Dinesh Bhatia gave their valuable insights about SNCI, Society for Neurochemistry, India. Finally, Mrs. N. Sudha, Assistant Professor, Department of Biochemistry, Ethraj College for Women, delivered the vote of thanks. Yesterday, the technical sessions were given by Dr. Saravana Babu, Professor, JSS College of Arts and Science, Mysore, and Dr. Dinesh Bhatia, Associate Professor, Department of Biomedical Engineering, Northeastern Hill University, Shillong. I'm very much certain that all the participants would have found both the sessions to be satisfying and informative. I once again heartily welcome the esteemed dignitaries, faculties, and fellow participants to the new Regional Virtual Symposium, NEUGLIA 2021, Day 2. I now call upon Dr. C. N. Deepa, Associate Professor, Department of Biochemistry, Ethraj College for Women, to introduce the guest speaker of the day. Wait.
Good morning and a warm welcome to all dignitaries and participants who have joined us on the second day of our symposium. The Department of Biochemistry, Ethraj College, aims to widen the knowledge of our students by taking the frontiers of education beyond books through guest lectures, workshops, and interactive sessions. Keeping this in mind, we have co-organized this regional symposium on basic insights of neuroscience against neurological stress with sessions by experts from different areas of neurological research. I take immense pleasure in welcoming Mrs. A. M. Mahalakshmi, Assistant Professor, JSS College of Pharmacology, Mysore, to deliver the lecture on demystifying the link between sleep and cognition. Mrs. A. M. Mahalakshmi completed her B Pharm from Rajiv Gandhi University of Health Sciences, Bangalore, and M Pharm from JSS University, Mysore. She is currently pursuing her PhD in JSS Academy of Higher Education and Research. She has attended 7 national and 4 international workshops and training programs and been part of the organizing committee for around 6 workshops and seminars. Ms. Mahalakshmi has filed a patent for an anti-stressor prepared with enriched blueberry, and she has published about 36 research papers and 4 book chapters in reputed national and international publications. She has received a grant, sum of rupees 50 lakh from DBT as part of public health and nutrition for her project on geriatric chronic cerebral hyperperfusion as a co-investigator. She has also received a seed grant of rupees 5 lakhs from JSS AHER for the project on chronic sleep restriction and also received SIG BBRC grant of rupees 1 lakh from JSS AHER for the project titled Investigation on the Role of AT1 Receptor Antagonism on Mitochondrial Dynamics in Parkinson's Disease. Mrs. A. M. Mahalakshmi was the recipient of Safety Pharmacology Society Junior Travel Award for the year 2011 and got the opportunity to attend the Safety Pharmacology Society meeting held in Innsbruck, Austria. She has also received the Best Poster Award in the international conference organized by SNCI on neurochemistry and neuropharmacology. She has also been conferred with IBRO APRC Fellowship. Ma'am, we are delighted to have you among us today. And I once again extend a warm welcome to you and request you to enrich our participants today Over to Mrs. Mahalakshmi.
Thank you, ma'am. I would firstly like to extend my thanks to Dr. Vanishri ma'am and the coordinators from the Etiraj Women's College for giving me a chance to deliver a talk. In spite of in middle of many specialized researchers like Saravana Babu sir, Dinesh Bhatia sir. I'm much junior compared to them. Like in 2019, I had presented a poster like under SNCI banner in one of the conferences which we have conducted, hosted in our institution. I'm very happy to present a talk under SNCI banner. In the neuroglia 2021, which has been organized by Etiraj College of Etiraj Women's College. Thank you, one and all. Moving on to the talk of the day, like I would like to give an overview or explain the link between the sleep and cognition. Firstly, why do we have to actually take sleep as so serious concern when compared to many of the deadly diseases today, like cancer and then the cardiac disorders? Is sleep is so serious to take it into as a main research stream or not? Like that confusion would have confusion will be there for everyone who starts the research with sleep. But over a period of time, when we actually see the deadly impact of the sleep or sleep restriction over a period of time that it is causing, it is definitely required to consider sleep as one of the upcoming research area. And I'm happy that we are also working on sleep and aging. Like I'm pursuing my PhD under Dr. Saravana Babu sir under sleep and aging. So linking the sleep cognition, my talk goes with the title Demystifying the Link Between Sleep and Cognition. It's like overall in the presentation, there are four different segments which I'll be giving overview on. Sleep and then the modern science is the first one. Like what do we know about sleep and what modern science has been telling about the sleep, that we will I'll discuss. And some of the molecular aspects of sleep and its and in conditions of sleep deprivation, what molecular changes have been reported in different scientific literatures. And our research finding, what we are doing in our lab, is one of the segment. And like what was the traditional medicinal science that says about sleep and the disorders or the impairments that the sleep restriction could cause. Those things will come as a last part. So overall, these are the four different segments of my talk. um, as we all know, sleep and wakefulness, these two are endogenous recurring behavioral states that reflect the coordinated changes in dynamic functional organization of brain that optimizes physiology, behavior, and health. This is the definition as per the National Institute of Mental Health, USA. Otherwise, in general, sleep, it is a reversible behavioral state of perceptual disengagement and unresponsiveness to the environment. How does the sleep evolved? See, initially, when we go back to the great apes and then the evolution of civilizations and all those things, sleep came into picture mainly in order to escape from the predator's risk. So the great apes, they used to construct the platforms at a relative height over the trees and all those things, so that like they could escape from the predator's risk. That was the major concern with which sleep came into picture. And like as they used to construct these platforms, the great apes could have they experienced a better quality of sleep and a longer duration of sleep. That in turn actually increased their cognitive ability. So the moment their cognitive ability was increasing, that made them to like refine their engineering skills in constructing the platforms more and more, like in a better way. So like as we understand, like if we compare like our own lifestyles from with respect to our grandfathers and great grandfathers, they never used to have like that huge mattresses or that huge cots at house. Like a simple mat, or like sometimes they used to sleep even on a empty floor also. So like the more and more sophisticated the platform, the more it is related to the cognitive ability, improvement in the cognitive ability. And that is how even the great apes, like they have developed more and more sophisticated platforms. So National Sleep Foundation suggests that 7 to 8 hours of sleep is essential for maintenance and restoration of good health. So like the sleep, it is not just the maintenance of like homeostasis. It is a means of like energy conservation, growth and repair, increased efficiency of metabolic process. All these things are linked with good sleep. Interestingly, humans are the shortest sleeping primates with more of flexibility. Like initially, the non-industrial civilization or the post-industrial civilization, if we compare that population, they were not having much of flexibility or much of variations in their sleep pattern compared to the post-industrial society which we are experiencing now. That might be because of socio-cultural, technological and lifestyle trends, which are the major contributors for these post-industrial variations in the sleep pattern. However, it plays an important sleep plays an important role in thermoregulation, homeostatic control over endocrine cascades, working memory encoding, and consolidation. So, like once these things gets disturbed, these homeostatic functions or the balance is disturbed, that could even lead to the imbalance in the circadian rhythm and the light and dark cycles. So there are like a quite relative terminologies like insomnia, sleep restriction, sleep deprivation, all these things. Insomnia is generally the inability to sleep adequately, either in length or quality, despite the opportunity to sleep. The person wants to sleep but is unable to sleep. So that might be because of the pain due to the illness. So such conditions will, when there is a pain due to illness or something like that, that will actually change the dynamics of sleep itself. So that is one condition. Secondly, sleep restriction. Sleep restriction is reduction in length of sleep time below an individual's usual baseline to maintain the optimal performance. It is like this is most prevalent in today's society. The reason might be many things: the addiction to social medias, or the gadgets, high use of gadgets, Blu-rays, all these things. It is going to be like the person's normal sleeping time for an adult male is like 7 to 8 hours, but then like every day 2 hours or 3 hours, 1 hour, if he is reducing, like maybe because they are watching a some movie or like something in the social media. So because of that, like every day 1 or 2 hours, if he is restricting, that might not be a serious concern for that particular day. But over a period of time, like when it accumulates for a chronic duration, if it happens, then that could also be a fatal. So that is what is sleep restriction, and like most of us are experiencing the sleep restriction in today's life. The third relative terminology is sleep deprivation. This is elimination of sleep for a period of time. It's a complete elimination of sleep. It can be like elimination of sleep for one night, for 2 days or 3 days, to significantly prolong the wakefulness. Like these conditions generally happen like during certain emergencies at home, like when there is a death or something at home, or like some trauma cases, or in case like students during before their examinations, a day or 2 days before the examinations, when they are spending time over for overnight preparations. Such things come under sleep deprivation. So these are some of the terminologies which comes. As I said, like sleep is not only to maintain the homeostatic balance, it is a means of energy conservation, growth and repair, increased efficiency. It leads to increased efficiency of metabolic process and all those things. So like when there is disturbance of this sleep, it do affect or it do disturb almost every system in the body. Amongst those things, like one important system is also immune system. So immune system, like it's kind of like it's a self-regulating system. When it gets disturbed, like it is a double-edged sword. Like it can be like sometimes when there is activation of immune system, it is like fruitful also, it is good also, but at the same time overactivation of immune system can also be a dangerous. So like when if you see this particular graph, like with respect to the time of sleep restriction or the types of this sleep restriction or sleep deprivation, how the immune or the inflammatory status is getting affected. So TSD is total sleep deprivation. When there is total sleep deprivation, it mainly affects the system by activating the innate immunity. That is like it increases the natural killer cells, number of monocytes and neutrophils. Thereby there will be increase in the C-reactive protein, and that too in fact increases the pro-inflammatory cytokines, and which also like leads to increase in the cortisol levels. So this is a acute phase, like in case of total sleep deprivation, when like we are losing sleep for a day or two, complete loss of sleep, the acute phase of inflammatory system gets activated. But over a period of time, like when the sleep loss has been continuing with sleep restriction, then like it is the adaptive immunity which gets affected. So here it is mainly the B lymphocytes which gets activated, and thereby again increase in CRP. The major pro-inflammatory cytokines like interleukin 1 alpha, interleukin 6, IL-17A, tumor necrosis factor alpha, all these things will be increased or released, and thereby the pro-inflammatory cytokines increases, and a chronic phase of immune impairment happens here. So this, like if at all, if there is a recovery sleep, then that recovery sleep may bring back the disturbed system to normal in acute phase. Whereas in case of chronic, it will be difficult, because like it's a gradual impairment that has happened. So this insufficient sleep recovery could lead to obesity, diabetes, cardiovascular diseases, and sometimes even lead to death also. So moving on to the sleep and the sleep cycles, as we all know, like there are two types of sleep: REM, that is rapid eye movement sleep, and non-rapid eye movement sleep. So rapid, like in a day or in a night, we can consider that like the sleep cycle, like they will be these are the stages of the sleep cycle with respect to the duration. First stage is the non-rapid eye movement stage. So this non-rapid eye movement stage will be for 5%, like the duration will be for initial few minutes. Then again second stage of NREM like takes up, that is for 45%. And then again third stage of NREM is for 25%. And after these three, a single phase of REM will come. So like this is one cycle. Usually one cycle lasts for 90 minutes, and like this there will be around 3 to 4 cycles happening in one night. And like the changes, the physiological systems which will be active and inactive during each phase of these stages are like this. So generally in case of REM sleep, rapid eye movement will be present, dreams will be there, muscle twitchings will be there, but all these things are absent in case of non-REM. When we observe the major physiological systems in the body, that is heart rate, blood pressure, respiration, body temperature, all these things are fluctuating in REM sleep, whereas in non-REM sleep they'll be under stable condition. So which indicates that all these systems will be still active. Okay, so because they are active, if there is like deprivation of this REM sleep or any disturbance to this REM sleep, whether it is in terms of like quality or quantity, both, that may lead to disturbances in any of these systems also. So this is in continuation with the sleep patterns which we discussed. So generally, like 10 p.m., like starting from 8 p.m. actually the preparation for sleep should start. But generally, the like average time, if we consider for the population to go to bed, it is 10 p.m. So from 10 p.m. to 6 p.m. if we consider, so there will be around 4 cycles of non-REM and 3 cycles of REM. You can observe here, like as the time is moving, proceeding, the REM sleep duration has been increasing. And as the duration increases, the quality of sleep also increases here. So this like considering this, like a study on young adults was conducted. So this is one of the data which we have taken from published literatures. So like in the young adults, like who have undergone like complete sleep, like they have not their REM or non-REM, nothing was disturbed, 100% REM sleep was there. So in them, you can see the rate they could memorize is so high. The learning ability will be high in them. In the similarly, like in the adults, like whose REM sleep was deprived to 50%, in them, like it is comparatively less rate of memorization. And then like if you observe the population where they had like 100% REM sleep deprivation, their learning ability and memorizing capacity is completely like lost. It is almost lost in case of these population. So this is evident even in the animal studies. So in one of the research paper, REM sleep and memory were like correlated. There was a direct connection which was revealed using the novel approach. Generally, the working memory is determined in the animals using a novel object recognition test. So the animals will be trained initially, like keeping two similar objects. They'll be trained. They will be the animal will be left for 10 minutes in that particular maze in order to get acclimatized to these objects. Similar objects will be kept here, like you can see two different objects or different colored objects are kept, but generally in the first stage the similar objects will be kept. And after that, after the training phase and the familiarization phase, on the like the animals were subjected to the sleep deprivation. You can see in this sleep deprivation, it is a like one night sleep deprivation only. You can observe here the sleeping pattern or the REM and the non-REM cycles. So together there should be 7 cycles, but you can see here it is not 7 cycles. It is just 3 cycles which is shown. So which means like there is a disturbed sleep pattern here that has made the animals not to explore the novel things. The general tendency for the mice or the rats is that like they explore the novel objects more. So when like similar objects are kept, like they'll be moving towards both the objects equally. But all of a sudden you remove one object from that and then keep a newer object, then the tendency for exploring the novel object would be more for the animals. But that tendency is lost here. So which means that like there is some disturbance in the consolidation of the memory, learning and consolidation of memory. So when we are talking about the memory, general stages in the like learning and memory goes like this. Initially, attention, that is listening, more of listening or learning is the attention. So from the attention, it is encoding. Encoding is like converting the labile memory into a more reformed form of memory or a like long-term memory. So the short-term memory, like which has been turned out or like which is shifted into long-term memory, is encoding. And then is storage. It builds up with the studying and use. The more we practice, the more we see, the more we learn, this like storage happens. And this completely and the next stage is retrieval. This retrieval stage will be completely dependent on the storage one, like how well the storage has been built up and how frequently it has been used. So based on that, the retrieval works. So in this pattern, like these are the four different stages of learning. And like when we are talking about this encoding here, this is the place where exactly the microanatomical structures and the synaptic transmission, they all play a role. So here you can observe like learning, it is the attention, and then consolidation is encoding and storage. It completely depends upon the slow wave sleep, that is the REM sleep mainly. The more this sleep is there, the more better is the sleep, the more will be the better consolidation happening, and that will lead to better retrieval also. So these are the different observations we have taken from different literatures, which clearly says that there is a direct connection that REM sleep alteration could lead to impairment in memory. So like in the memory, as I said, like from the sensory registration or from encoding, through perception, a short-term memory will be formed, and that short-term memory will become long-term memory. And like from the long-term memory, the retrieval happens. So from the retrieval, the working memory can be checked actually. So this is a process phenomenon. To explain that in a like anatomical way, more microanatomical way, the structures which are involved in establishing this particular pattern of learning and memory is the dendritic spines majorly. So dendritic spines, these are the very small projections or protrusions from the dendrites of the neurons. They will be of different shapes. You can, if you observe here, like there will be a cup-shaped structure, and then there is a neck which links to this shaft. The middle one is, this one is the shaft of the dendrite, and to the shaft of the dendrite, both the sides, there will be different types of spines that are formed. So these dendritic spines are the prominent sites for the synaptic transmission. So the more of the synaptic transmission happens at these sites. So there will be different types of dendritic spines like philopodium based on their shape and the size. Philopodium, thin spines, cup-shaped spines, stubby spines, and mushroom-shaped spines. These are different spines. So like when these transmissions, as I said, they are the prominent sites for the synaptic transmissions to happen. So when the synaptic transmissions are will happen at these dendritic spines, they can either lead to a long-term potentiation or long-term depression. Long-term potentiation is something like strengthening the synaptic plasticity. So this, whenever this happens, LTP happens, there will be increase in the dendritic spine number, expansion of size of the head of the spine, conversion of thin spines into stubby or mushroom spines, stabilization of newly formed spines happens, and increase in synapse number and strength and neural connectivity also. So it's a good thing. LTP is a good thing overall. And LTD is long-term depression. So whenever this LTD occurs, like that leads to decrease in the dendritic spine numbers, shrinkage of size of spine head, destabilization of the existing dendritic spines also could happen, and decrease in the synapse number and weakened synaptic connectivity. So one increases the neural connectivity, the other one decreases the neural connectivity. We have observed that like in the literatures, we can clearly see that like most of the types of sleep deprivation or restriction cases have induced LTD mainly, which means like it has gone for the spine shrinkage. So LTP like increases the spine number as well as the size, so spine enlargement, whereas sleep deprivation or restriction is said to cause LTD, which means like there is spine shrinkage. So this spine shrinkage, and with that like lack of sleep, as we know, like causes, we have understood that like microanatomically it is affecting the dendritic spines, and thereby like major physiological phenomenon, that is memory is also getting affected through the impaired neurosynaptic transmission. So we know different parts of the brain, they actually are like integrating different concepts or different functions. So lack of sleep can cause parts of our brain to slow or shut down completely. For example, like temporal lobe, we know like it encodes for language. So prefrontal cortex is majorly for judgment and visuals, and frontal lobe encodes for creative thinking. Parietal lobe encodes for math and logic. Neocortex encodes for memory and learning. And in the neocortex is the major like organs for the memory and learning come like hippocampus and all these things. So like whenever there is a sleep loss, like neocortex is majorly affected, and that will lead to difficulty in learning the new tasks and making novel connections are seen or observed in the literature. So whether the similar kind, like that was in the humans, like what I have explained, like hippocampus is being the major area for like learning and memory, will that replicate even in the animals? So to show that, like the animal research or sleep research in animals, there are like quite a few experimental models. Like generally the animals will be like, when we receive, we leave it for acclimatization, and there will be induction of sleep restriction for a period of 21 days. This restriction, like sleep restriction, was for 21 days, and like generally 6 hours is a like standard protocol which we use. And as the animals are nocturnal, these animals are nocturnal, mice and rats, like the sleep deprivation will be usually restriction will be usually induced during daytime from morning 6 to 3 o'clock. So like from week 1 to till day 21, there will be day 0 to day 21, there will be sleep restriction happening. And then at the end, there will be sacrification, biochemical estimation, and like some tissues will be sent for histopathology, and protein studies will be performed. So like when a study has been conducted like that, like a normal animal which is not subjected to sleep deprivation, that has shown a systemic approach in learning and memory. That is also, you can also see that in this particular diagrammatic representation, wherein the shape of the hippocampus or CA1 DJ regions are also shown here. So the pattern of learning and memory is clear in the normal animals, while in the animals which are sleep deprived, you can see there is this is a disturbed pattern of learning and memory. So it is not organized. And at the same time, the treatment group here, it shows that on treatment, the treatment can bring back the learning and memory to a normal form. So this is one observation which we have taken, which was like much connected to the theme. And then the different types of sleep restrictions can be used in animals. There are different models, such as gentle stimulation is one, wherein like you entail tapping or moving the cage of the animal. Like whenever the animal is tending to fall into a sleep, you tend to like move the cage, or like tapping on the cage, so that like you are disturbing the sleep. So that is one way, but it is a little laborious one. And second one is a forced locomotion one. Like you have to design an apparatus in such a way that like when you keep the animal on that, like it has to be constantly on motion, so that it doesn't the animal doesn't tend to fall asleep, is the second model. This is also like quite laborious, and also the like muscular fatigue may happen in this case. And the third one is the flower pot method, or it is also popularly known as modified multiple platform method. So in this method, what we do is these are the models to induce sleep restriction in the animals. In this particular model, what we generally do, and this is the method which we have employed or stabilized in our laboratory also. So in a cage, we fill water till 1 centimeter, and on that, in the cage itself, like we keep some cylindrical rods. The rods might be made up of like metal rods. So like the diameter of the metal rods will vary for rats and mice size. So whenever, I mean, the rods will be placed in such a way that like they should be enough distance between the rods so that the animal can move from one rod to another rod, but it should not stretch its limbs and start sleeping. So the distance between the rod should be maintained in such a way. And like whenever like you keep the animal on the rod, the animal tends to fall asleep. So when it falls asleep because of muscle tonia, the animal tends to fall into the water. So once when it falls into the water, then it comes back and like stands again on the rod. So like this is how the animal's sleep will be disturbed, and this continues for almost 21 days. Morning we keep the animals in the cage with the rods, and then by 3 o'clock like we remove it and we put it back into a normal cage with the husk and all those things. So every day this procedure continues, and then the sleep restriction would be induced in the animals. So like animals sleep restriction is induced in that way. How do we actually estimate the like memory in these animals? The memory can be determined either like both spatial and working memory both can be determined. Generally spatial memory is like determined in Morris water maze. Morris water maze, it is a tank. It's a big tank which is filled with water, and like the whole of this circular tank will be made into four different quadrants. You can see in this particular image that the circle is being divided into four quadrants. It is the same image of this maze, water maze, which has been divided into four different quadrants, and the quadrants will be named as like southeast, northeast, south, northwest, and southwest. And in one of the quadrant, the platform will be kept. So below the water a platform will be kept. And like this experiment is for 5 days actually, like 6 days. 5 days training will be there, and the sixth day there will be a probe test happening. So 5 days of training means like first day will in one of these quadrants we will leave the animal. Generally if you can observe here, like here they have left the animal. So when we leave the animal here, the animal goes in search of a safer place. So when it is going for in search of a safer place, like when it finds this platform, it will actually feels that it is a like safer place, and it will be like sitting on the platform itself. So this procedure will be done for 5 days. So initially first day it will take more time, second day like little faster, so that like when we reach the fifth day it will clearly know that like this is a place where the platform is kept. And in order to facilitate that, some of the visual cues will also be kept. So like some colored cues will also be kept, and the whole of this thing will be recorded from the day one till the probe test. All the like procedures which we are doing will be recorded. And when we like compare, in the last day what we do, like we remove the platform and then we'll allow the animal to search the platform. So generally the normal animals which are not induced with sleep restriction, those animals will go immediately to the platform region area, and then like they cannot find the platform, so they keep searching for it in the same place near to the same area. However, the sleep induced or sleep restriction induced animals, those animals will also go to that place, but then like they will not search more there. Like they will be like throughout the maze, they'll be roaming and they'll be searching for the platform. So that difference will be clearly observed in this Morris water maze test. We can say this is a golden standard for measuring the spatial memory, determining the spatial memory in case of especially the sleep restricted animals, because it is also a there is also a stress applied here. The animal is not left free. If the animal is left free in a space, then it may tend to sleep. As here, like there is water and it has to swim, it cannot like stay at a place. Then like the animal moves and then it exhibits the like activity very well. So the same has been like recorded and it is shown in these images. So like in case of normal animal, you can see the animal which is left in this quadrant has traveled. This is a quadrant where the platform was kept, and you can see like how well it has explored the platform area. While in the sleep deprived animal, you can also see here, like the animal has gone through this area or the quadrant, but then at the same time it has roamed around throughout the maze. So similarly, even like the time taken to reach the platform, so the time taken to reach the platform is the escape latency actually. So that initially in the starting first training day, you can see it is same for both the non-sleep deprived and the sleep deprived animal, but like it went on decreasing for the normal animal, non-sleep deprived animal. It went on decreasing compared to the sleep deprived. So sleep deprived, you can see in the red color, like the duration has been like more only when compared to normal animal. So the time taken to reach the platform is also more in case of sleep deprived case. So this is one of the experiment which we are part of, one video of the experiment which we have conducted in our lab. So the animal was kept left here, and then you can see how well the animal went and then explored its platform, and it is sitting there like it is during training phase. While during the probe test last day, this platform will be removed and the animal is asked to search for it. Okay, so the same, you can see the total distance traveled by a normal control. I mean, the normal animal is very less, while sleep deprived animal has traveled a lot of distance, a long way. Like it kept on like searching, and the same like is seen here. It is a track plot. All these movements will be recorded. So each of this area, these recordings will be analyzed using a anime software. That software gives us the images like this. These are called as track plots, and through the track plot also we can, like, this is visually understanding that how well the animal has reached the target quadrant, and then like in sleep deprived group, how the animal has taken a long way to reach the target quadrant. So in a similar way, working memory can also be assessed. That was for the spatial memory, Morris Water maze. Working memory can also be assessed using novel object recognition test. So here in this, like there will be three different phases in the test. It's a 3-day procedure. First day, it is habituation. So there will be a maze. It's an open field kind of maze. Like in the it will be divided into again many quadrants, and then in this particular cage, the maze, the animal will be left free in order to habituate itself to that particular area. All of a sudden, if we bring an animal to a newer environment, it gets anxiety, and then like it feels that it is not safe. So that anxiety related issues will be more rather than like what we want to actually check. So like in every experiment, if we could see, there will be a habituation, training, familiarization periods to make it comfortable to the procedure that we are doing, and then they are trained, and finally it is tested. So first part is habituation. It will be for 5 minutes. The animal will be left empty. It will roam around. It will see, like try to accommodate itself in the maze. Next, second day, the same animal will be left with two different objects placed opposite to each other. So these two objects will be the familiar objects. I mean, the similar objects are kept, same color, same dimension. They'll be of same type of material with which they are made of, and this phase is called familiarization. It will be for 10 minutes. So like because of the order and then the texture of the objects which we are using, the animal gets itself familiarized to those objects. And then the third day is again the testing day. Here, like one object will be replaced. You can see like two squares were there here. Like here, like one square is replaced with a star, so different object will be kept here. So if it is a normal animal, if its memory is normal and if it is working normal, then the animal needs to actually explore more towards the novel object. But like this procedure, this as clearly shown in the results, that in the sleep deprived cases, the animal is not exploring the novel object. Like instead, it is like moving similarly with both the objects. So this clearly indicates that the sleep deprivation also, or restriction also, affects the working memory along with the spatial memory. You can see the reduced discrimination index in case of sleep deprived group. So this is one of the image from the microanatomical background. So as I earlier said, like there will be whenever there is sleep restriction, there will be induction of LTD, and that LTD will reduce or causes the shrinkage of these dendritic spine or loss of dendritic spines. So this image, like this is from our lab only. So in this image you can see in the control animals, normal animals, the density or the and then the length of the dendritic spines from the shaft here, dendritic shaft here. And at the same time in the sleep deprived group you can see like the density of the spines, and at the same time the length of each spine also is reduced. There is shrinkage of the spines happen in the sleep deprived group. So the similar image is this, the sleep and dendritic spines are decreased in this particular image. And at the same time sleep deprivation not only affects the microanatomical structures such as dendritic spines which are involved in the synaptic transmission, but also reports have shown that like it can also affect the neurogenesis. Generally the neural stem cells from the dentate gyrus region, that is the granule cells, they develop into the neurons. So like that is what is called as neurogenesis. And you can see here it's a normal group. A is a normal group. So from the normal group in the dentate gyrus, the number of these neural stem cells are more. These neural stem cells will develop into the neurons. So the number of neural stem cells are more here when compared to sleep deprived group. This is a sleep deprived group. In the sleep deprived group there is like a reduction in the number of neural stem cells, but the same thing on treatment, the number of stem cells have been increased. So there are reports, these reports also clearly suggest that like it not only affects the synaptic transmission but also the neurogenesis, which itself is a basic phenomenon for the neural system to work appropriately. And this is again with respect to the synaptic transmission itself. So whenever there is normal sleep, in during normal sleep the synaptic transmission also happens in a normal way. There will not be any disturbances in the different postsynaptic markers and the presynaptic markers. Here we can observe in case of sleep restricted animals, they can it can be either due to increased interstitial flow, increased magnesium, increased calcium, or decreased levels of potassium. All these things may contribute to affect or to impair the synaptic transmission. And this has been also evidenced through the research findings from our lab. In the sleep deprived group, like we have estimated or determined the both the presynaptic protein levels and the postsynaptic proteins in the in normal and then the sleep deprived animals. So presynaptic proteins like postsynaptic density 95 PSD and synaptotagmin SAP 97, you can see in case of normal, these plots are for the normal animals and these are for the sleep deprived animals. So in the sleep deprived animals there is a drastic decrease in the protein levels of the presynaptic proteins. At the same time, even with respect to postsynaptic proteins like synapsin is also reduced in the sleep restricted group. Not only the presynaptic and the postsynaptic proteins, there is also decrease or the down regulation of the neurotropic and the other growth factors like PCREB is also reduced, BDNF is also reduced. Having said that, like when there is a drastic down regulation of PCREB, BDNF, presynaptic proteins and the postsynaptic proteins, that like ultimately affect the synaptic plasticity. So apart from the microanatomical and then the image findings, even the histopathological findings also suggest that like in case of sleep deprived group, there will be like neurodegeneration happening. So you can see this is a control group, like there are like numerous neuronal cells present here, while at the same time in the SD group, sleep deprived group, there is the number of intact neurons are reduced. So currently, those are all the findings. Like a few of them are from the published literatures, and like most of it from our lab. So our lab is focusing currently on at different at different levels of the signaling pathways that are involved in the sleep and sleep deprivation. So the major ones are know that like during sleep deprivation, GLP-1 and then the PDE4 are like major proteins, and then when there is like increased upregulation of like PDE4 enzyme, that can result in like decreased cyclic AMP, which means like there is more breakdown of 5-AMP into cyclic AMP. So the quantity of the amount of cyclic AMP is reduced. So cyclic AMP in turn is related to many of the signaling, or linked to many of the signaling cascades which regulate the synaptic plasticity, LTP, and memory. So that can be through like PKA through the CREB, or it can also be through the general neurotropic factors and growth factors like BDNF, all these things. So our lab is mainly working on now like on the PARP and histone deacetylase enzymes, that is sirtuins and PARPs, in order to increase the glucosid protein, and then on also on working on the PDE inhibitors in order to like increase the cyclic AMP level. So thereby, by doing these two, there can be like increase or the normalization or restoration of the cyclic AMP related signaling cascades which were disturbed during the sleep deprivation. So this is one of our paper, like where we have collated or summarized the observations on sleep, brain vascular health, and aging. So in this particular thing, like we had seen many things on sleep correlating to sleep restriction and deprivation correlating to the memory and cognition, but also there is a lot of evidences which we are observing that like sleep restriction can also cause prominent changes in the brain vascular structure and even function. The elasticity, loss of elasticity in the vasculature, and then like even with respect to the markers like ICAM, VCAM, and all these things, and that that can also lead to the preponement of aging, which has been actually correlated in this particular paper. And that the paper also in the paper we have also shown that like even sleep deprivation will affect the mitochondrial changes. And we can see in the normal animals the mitochondria will be generally the oval shape with the intact cristae and all those things, while in case of sleep restricted animals, deprived animals, we can see the oval shape getting turned into a circular shape. So you can see those images clearly here. So these are best examples that like sleep is not so such a thing that like it is only causing some prominent like visual effects, like if there is a sleep deprivation, some accident happening on the next day and all those things, but also it's kind of a slow poison. Every day it is inducing a change in our anatomy and the physiology, so that ultimately like it is been brought up as a like turned out to be one of the major complications of health. And one more paper where we discussed about sleep and the effect of sleep loss on different body fluids. So this is again one paper from our lab where, as I said, like we were working on phosphodiesterase 4 enzymes as a therapeutic target in neurological disorders. Like how by by inhibiting this phosphodiesterase 4, the cyclic AMP can be restored, and through which like the downstream signaling cascades would be restored. So with respect, that was a this was a review paper, and the research paper from our group on the same theory states that like this is a cell line study wherein like the similar to sleep, we cannot create a condition in the cell line, so we use it quinolinic acid as a which is excitotoxin, which actually decreases the cyclic AMP. So this we used to induce a condition similar to that of sleep, but which cannot mimic the physiological sleep loss condition. But when this quinolinic acid treated cells have been treated with rofilumlast, there is a significant restoration of the cyclic AMP, and which has been shown through this paper. So, with all these things, having discussed all these scientific literatures and then our lab findings and all these things, like there are multiple pathways and multiple signaling cascades which are getting disturbed in the sleep restriction. So, to restore those things, the major working majorly on GABA neurotransmitter and GABAergic pathway may help to restore all those functions. But at the same time, there are like many limitations to work on GABA, because like most of the drugs available in the market, which are of the GABA inhibitors and all those things, they are all habit forming ones, and like a long term usage is not advisable. So it is also it is better to work on something which is a lifestyle modification, and which is from a nutrient point of view, which can restore these sleep restriction related effects on like memory and cognition. So as I said, our lab is working on sirtuin and PARP pathway, PDE4s, and another one important pathway which we are concentrating is on tryptophan metabolism. Tryptophan metabolism, tryptophan is a like basic amino acid that is required in the body, and this tryptophan on metabolism, it gets one pathway leads to the serotonin formation, the other pathway leads to formation of kynurenic acid and quinolinic acid, wherein kynurenic acid is a neuroprotective while quinolinic acid is a neurotoxin. So working to increase the amount of kynurenic acid will also improve the memory and cognition. So brain foods we started working on, like one of the important food in that category is the blueberries. So these blueberries are rich in polyphenols, as we know, they contain phenolinic acids, flavonoids or non-flavonoids, all these things at different these things. Phenolinic acids have hydrobenzoic acids and hydroxycinnamic acids. Flavonoids, there are different types, anthocyanins and anthoxanthins. And then again, under anthoxanthins, there are like flavonols, flavones, flavonols, isoflavones, and flavonones. Non-flavonoids, there are stilbenes and lignans. In that, like one important thing, stilbene is resveratrol. So triphala is also one such agent, one nutrient supplement from the Ayurvedic side. The word triphala is named because its formulation has three fruits, namely haritaki, bibitaki, and amalaki, like which are classified and rasayana drugs. So this triphala formulation is a rich source of antioxidants, frequently used to treat many diseases such as anemia, jaundice, constipation, asthma, fever, and chronic uses. It's a even if not to treat the diseases, it's a very good antioxidant immune booster. So generally people take this on a daily basis. So keeping these Ayurvedic things in mind, we designed the study objectives to study the effects of this blueberry on sleep restricted animals, cognition and biochemical paradigms. So also to study the effect of this blueberry extract on kynurenic acid content in brain of the sleep restricted animals, and finally on brain antioxidants. So, like the animals used were like Vister albino rats, the animal approval number is also mentioned. So, the sleep restriction is induced by multiple platform method, and then the treatment doses here we have taken. The blueberry extract, low dose and high dose, and then there is this triphala taken as a standard reference. So it was a chronic study for 21 days. The sleep restriction was induced for 8 hours every day from a day one, and like simultaneously the treatment was going on 22nd day. On 21 second day, memory evaluation was done through step down latency and followed by necropsy and the biochemical and the protein estimations. These were the groupings: non-sleep restricted group, sleep restricted group, and then the polyphenol enriched blueberry, low dose, high dose, triphala, and drug control polyphenol enriched blueberry again. So the after 21 days of treatments, they were like checked for the behavioral paradigms, mainly memory on step down latency, and then biochemical parameters like brain kynurenine level, and the antioxidants like SOD, GSH, and LPO were also treated. So the sleep restricted animals, you can see there is a lot of time taken. They have taken less time to move from these to step down through the step down latency. Excuse me. And through lipid peroxidation also shows that the sleep restricted group have are showing significant increase in the lipid peroxidation rate, and at the same time the treatment groups have reduced. In case of superoxide dismutase also, there is reduced superoxide dismutase level in the sleep restricted group compared to normal, and the treatments have increased. Glutathione also is reduced in sleep restricted group, and it is increased on treatment with this polyphenol enriched blueberry extract. So on kynurenine levels, the kynurenine level is also reduced here. This might also be correlated to, like, there might be more conversion of this kynurenine than to kynurenic acid from to the quinolinic acid. So that might have led to decreased level in the kynurenine. And through DPP assay, again, it is shown that like the is not the percentage inhibition is increased in case of sleep restricted group, and FRAP assay also suggests the same. So overall, like in case of the polyphenol enriched blueberry extract has restored the kynurenine. So like after the treatment, when we calculate there is restoration of kynurenine in case of treated groups. And similarly, the histopathological findings suggest that there is like deposition of beta amyloid. This is a Congo stain of the CA1 and DG regions of the hippocampus. So there is more this thing of Congo red taken up here, like because of the more beta amyloid being taken up the Congo red, that is indicating in this particular region. And on treatment, you can observe like low dose and the high dose is showing less less beta amyloid markers here. And yeah, so that was the literature, I mean scientific output from our study, like how the nutritional supplements can also enhance memory and to overcome the sleep restriction induced memory impairments. So like the exposure to blue light has been like kind of, it is very much ignored, but the consequences of it is not kind of like we cannot ignore it. uh, People whose melatonin levels are suppressed and whose body clocks are thrown off by light exposure are more prone to depression. So, because of the blue rays that are thrown by these gadgets, there will be significant alterations in the melatonin level, and that can lead to depression. And that in turn can lead to the buildup of neurotoxin, because like they might be increased in the beta amyloid levels, and then the glial system will not be working appropriately. So because of that, the buildup of neurotoxin also increases, and this can lead to a poor night's sleep caused by smartphone light, can make it harder to learn, so that it can, like all these things together can impair memory also. And other than this, blue light also affects retina of the eye, so the incidences of cataracts are increasing, and reports also suggest that the blue light or blue rays induced breast and prostate cancers are also increasing. And obesity is another general lifetime lifestyle related disease which has been happening because of more exposure to blue light. So to all those things, the last part of the content of the my talk is about the traditional medicine. See, all these importance of sleep is not being, like, though more scientific evidences are being given nowadays, but then like the importance of it has been established 10,000 years back by Shushruta, Acharya Shushruta. He says that the pleasure and sorrow, the nourishment and malnourishment, strength and loss of strength, libido and post-sexual virility, good intellect and poor memory, pleasure and sorrow of full living are all dependent upon proper and improper sleep of an individual respectively. So like they had categorized a whole day 24-hour cycle into different segments here. This is an Ayurvedic clock which says that like it is mainly dominated by three doshas, that is vata, pitta and kapha. And like in this particular thing, if we can observe at every point of time, like in a day and the night, one particular dosha will be like it will be active. So like when that particular dosha is active, certain activities are set to do during that particular period, and certain activities are not allowed to do. So because if we do the activities which are not suggested, then there might be change in the doshas that could happen. So it is the alignment of our daily rhythm in tune with the nature's rhythm. So support your day by the dominant energy inside and around us. Each day we cycle through the three doshas. The day is divided into 6 4-hour cycles. So you can see here like 1 2 3 4 5 and 6 4 4 hour cycles, 10 a.m. to 2 p.m. and 2 to 6 p.m., 6 to 10 p.m. again. So starting from the dawn, from 6 a.m. to 10 a.m., if we can see, kapha will be prominent here. And what are the activities that we can do? Exercise, eat lightly, elimination completes, and prepare for the day, gather energy for the day. So these are the things that we have to do. And at the same time, if we observe like this dusk period from 6 p.m. to 10 p.m., you can see assimilate, slow down, prepare for sleep. 8 a.m. they're telling like prepare for sleep. So at 8 a.m. we should start preparing for sleep, go within, and by 10 p.m. there will be deep sleep. And like throughout, like you can see midnight there will be pitta, and then like morning, like when from 2 a.m. to 6 a.m. there will be vata, and this is the best time for like having light sleep, meditation, visualization, creation, and all those things. The Brahma Muhurta, which we call, that falls in this particular period, in between 2 a.m. to 6 a.m. That is around like during that the vata will be prominent. So yeah, it makes sense to sleep well within 10 p.m. because kapha is dominant and it is related to sleep. Sleep beyond 12 a.m., pitta dosha will be dominant. So not only sleeping will be uncomfortable, it also makes prone to headache, gray hairs, metabolic diseases, early aging, etc. So human optimization pyramid says that like sleep is a base for all the activities or for all the dynamics of the human life. So sleep, appropriate nutrition, good fitness enhances cognition. Cognition can enhance happiness, and that in turn can increase the lifespan. So overall, on a whole, I would like to conclude by telling that sleep is an indispensable component of like overall health and well-being. It not only affects the physical health but also mental health. In practice, in particular, it is cognition or cognitive function by triggering various pathologies like impaired neurogenesis, decreased spine numbers and structure and the function, like impairment in LTD and LTP, synaptic plasticity changes, and finally leading to cognitive impairment or declining. So, like on a whole, sleep deprivation impairs learning, consolidation, retrieval, and synaptic plasticity. So it is necessary to give a time to sleep and have a good sleep, which in turn is correlated to healthy life and well-being. So these are our products which are developed from our lab. One is polyphenol enriched beverage as anti-stressor. Patent has been filed for this, and technology transfer is in process. And tannin-rich natural product syrup for gastric ulcer management. So, both of them are developed based on the gut microbial function, and that been correlated to the cognitive enhancement. So that is also one of the area which our lab is working on. Yeah, finally, this is our group, Brain Behavior and Cognitive Research Group, which is headed by Dr. Saravana Babu, sir. And I wholeheartedly acknowledge, sir, for all the support that he has been extending, and for my thanks to Dr. Vanishri ma'am and the coordinators from the Etiraj Women's College for giving me a chance to deliver this talk on this SNCI platform. Thank you. Thank you very much.
Thank you so much, ma'am, for giving us a quality lecture. We'd be really happy if you could clarify some of our doubts, ma'am. Ma'am, the sleepwalking have
Sorry, ma'am, the voice is not clear. Can you please repeat?
yes, ma'am. We'd be really happy if you could clarify some of our doubts, ma'am. The first
Yes,
question
please,
is
please.
yes, ma'am. So, does sleepwalking have any neurological impact on our body, ma'am?
Speed walking,
Sleepwalking, ma'am. Does it has any?
sleepwalking,
Yes, yes.
sleepwalking on memory.
On the
You're telling
does it have any neurological impact on our body, ma'am?
neurological impact on body. See, sleepwalking, if it is in a normal sleep, I don't think so. It will have any neurological impact if that has been disturbed, because sleepwalking, it's a natural phenomenon that will be there like in the REM sleep. So if it is normal, there is no effect on the neurological findings as per my understanding.
Yes, ma'am. Yes, ma'am. And the second question is, does sleep pattern reflect on the memory and the learning process, ma'am?
Yes, definitely, definitely. REM sleep, though the time or the duration is less for REM sleep, it is very much important, and like most of the learning and then the encoding that happens, consolidation of memory happens during REM sleep. So the patterns are like very important.
Yes, ma'am. And the final question, ma'am.
Yeah.
The reading mode which is available in the smartphone, is it better than the normal blue light, ma'am?
Sorry, the
The reading
re-
mode that is available in the
reading
smartphones, ma'am, is it better
reading
than
mode.
the yes, ma'am? Is it better than the
Uh,
normal blue light, ma'am?
I don't think so. Firstly, the gadget itself is not appropriate for reading. Like it is always better to read with a book or having a paper on your hand. That is how even the field of learning also happens. And but I don't have much idea on like the reading mode, what type of blue light that it actually gives out when compared to the normal blue rays. I don't know whether it is totally different from the normal Blu-rays that the gadgets eliminate.
Yes, ma'am. Thank you, ma'am, for patiently answering all our doubts. And thank you once again for enlightening us with the insights of the topic. I'm sure all the participants would have found the session to be really informative. Thank you once again,
Thank
ma'am.
you. Thank you. Thank you.
It's now time to move on to another stupendous technical session. I now request Mrs. Tamar Ovia, research scholar, University of Madras, to introduce our next guest speaker.
A very warm welcome to one and all present here. In addition to invited lecture
as erstwhile student and as a member of local chapter member, I cordially invite and I'm very pleased to introduce Dr. J. Jayalakshmi. Dr. Jayalakshmi, she is the guest lecturer, Department of Biochemistry, University of Madras, and she did her PhD in the University of Madras. Her area of specialization is glioma. She has qualified the SLET exam for lectureship, and she has good publications in high impact factor journals like Springer Elsevier. And she has also published her research works in national and international conferences, and she has received Best Paper Award. And with these few words, I call upon Dr. J. Jayalakshmi to deliver her lecture. Over to you, ma'am.
Thank you. Good morning, all and all present here. Before I'm going to deliver my talk, I would like to express my sincere thanks to my guide, Dr. A.J. Vanistry, who is giving me this opportunity. And also, I would like to extend my thanks to the Etiraj College and an SNCI for giving me this platform. And also, I would like to say a few words regarding my experience with SNCA. I'm happy to share this. This is my third presentation in SNCA, but this is the first presentation as a speaker. Before that, I have presented my work as a research scholar, but I'm happy to say this. This is my first experience to speak as a speaker. With these few words, I would like to start my presentation. The topic today I am going to present is an adaptation of a cancer cell to the hypoxic stress. Hypoxic stress is also a kind of a stress that is faced by an cancer cells, especially in case of a solid tumor. Glioma, it is one of the solid tumor. It is facing this stress by adapting itself towards the metabolic reprogramming. This is a short research work. This is a simple topic, and this is going to explain only about how the cancer cells that is facing this stress and how it adapt itself for an energy metabolism. Hypoxia is nothing but an oxygen deprivation condition. Okay, whereas in case of hypoxic tissue, these tissues are inadequately oxygenated. Okay, especially in case of cancer, there is an abnormal formation of the cells. You can see the solid mass of these tumor. You can see then abnormal growth of the cell, and we find an disturbed growth of these cells, and there is an these cells that are growing apart from these blood vessels are deprived of an oxygen. Okay, you can see the cells, these cells that are nearer to these blood vessels are all highly oxygenated, and these cells are an aerobic cells, and the cells that are little far apart from these are aerobic cells are called an hypoxic viable cells. These cells are in an hypoxic condition, but it is still viable. Okay, and this is what the last this necrotic cells, this is very far away from the blood vessel, so there is a lack of oxygen and these cells undergo a necrotic death. Okay, today we are going to see about how these hypoxic cells are adapting for their survival even in the absence of oxygen. Okay, hypoxic stress, it is nothing but a stress. This is a consequence of an uncontrolled tumor growth. This is not a cause for a tumor, but this is a consequence of an uncontrolled growth. These might result in the structural and functionally disturbed microcirculation. This lead to an imbalance between the oxygen supply and then consumption. Okay, and these hypoxic stress is the major driver for the cancer progression. And what are all the influence of these hypoxia on cancer cell? Hypoxia, which the major influences, it diminishes the ATP production. Okay, by lowering the activity of the electron transport chain, and obviously there is a decrease in the respiratory rate because of an electron transport chain only. It utilizes the oxygen for an glucose oxidation. Okay, if there is a lowering in the activity of electron transport chain, there is a decrease in the respiratory rate of the chain. Then how come the cells that face the energy demand, there is a no ATP production, there is a decrease in the ATP production through electron transport chain. Okay, and then there is a decrease in the respiratory rate. Then how the cells which face the energy demand of the cell, how the cells it adapt to this stress. And also this is a tumor cell, it uncontrolled, it get proliferated uncontrollably, so it need a more energy. So how these tumor cells it survive and proliferate in this hypoxic stress condition, this is what the question raises for this research problem. Then metabolic reprogramming, it is a newly recognized hallmark of the cancer. Okay, this is what the major hallmark, and this plays major role in the energy metabolism by directly supporting the rapidly control cell growth. Okay, and hypoxia, this plays major role in the metabolic reprogramming of these cancer cells by activating or increasing the cell demand for the synthesis of biological molecules, and it plays major role in progression and also in the treatment resistance. Now we are going to see how these hypoxic stress that plays major role in adaptation of the cells for an energy demand. We can see the energy production in a normoxic condition, that is nothing but a figurative explanation for the energy production in normoxia and also in case of hypoxia. In this figure you can see the glycolytic cycle which take the sugar, that is a glucose, it takes the glucose. Okay, and then this glucose get oxidized into, it is converted into pyruvate, and this pyruvate which enters the TCA cycle where we produce two molecules of AT, and this pyruvate it enters the TCA cycle and it gets the NADH, and it reduces the NADH and FAD. These NADH and FADH enters the electron transport chain where in utilization of oxygen, these get again reduced and results in the production of an ATP. In case of normoxia, this is for the normal energy metabolism of a glucose, whereas in case of hypoxia you can see there is an increase in the utilization of the glucose when compared to that of normoxic condition, and you see there is a disturbance in the electron transport chain and also even in case of TCA. So the major energy production is mainly depend upon the glycolysis. So glucose is converted into lactate and it never enters into the Krebs cycle and results in the production of a ATP, but it is very less when compared to that of an oxidative phosphorylation. This is what the bypass way the cells in hypoxic condition it adapted for the energy production. Okay, if an adaptation, if that is the major focal point which plays major role in adaptation, this affects the this if one, it is nothing but an hypoxia inducible factor 1 alpha. This is a transcription factor which plays major role in expression of specific set of genes that are involved in the energy metabolism. These F1 which targets the genes that encoding the glucose transporters, glycolytic enzymes and then lactose dehydrogenase. In addition to this, these F1 is if one which plays which act as a resistor for the mitochondrial metabolism by limiting the entry of the pyruvate into the TCA cycle through the upregulation of the pyruvate dehydrogenase kinase 1. This kinase it phosphorylates the pyruvate dehydrogenase and results in the blockage of conversion of a pyruvate into acetyl CoA. So this pyruvate this pyruvate enters the conversion and converted into lactate through LDH, and this is the major mode of the energy production in case of hypoxic condition. So this if one activates these enzymes in the glycolytic cycle and it plays major role in the adaptation of the hypoxic stress. Anaerobic glycolysis, it is nothing but an tumor cells, especially in under the hypoxic stress, which undergo a significant shift from the oxidative phosphorylation to the anaerobic glycolysis. Okay, the cell which is mainly depend upon the glycolysis for its ATP production. Okay, so through the GLUT and then glycolytic enzymes. So overexpression of these PDK and then GLUT and then glycolytic enzymes plays major role in adaptation of cells towards the hypoxic stress through anaerobic glycolysis. This is for the figurative illustration of the energy production in case of normoxic cells and in case of an hypoxic cells. In normoxic conditions, the most of these pyruvate it enters into the mitochondria where it enters the oxidative phosphorylation and results in the production of an ATP molecules. Whereas in case of an hypoxic condition, only 5% of these pyruvate enter into the mitochondria and it enters the TCA cycle. The remaining 85% and enters into the lactate production. This is what the major adaptation that takes place in the glycolysis. And even though in the presence of the uh, glucose, the cells which this is for the tumor cells, and this mainly depend upon the lactate for the energy production rather than entering the TCA cycle. It prefers lactate as a lactate production rather than complete oxidation of the glucose because of the absence or absence of the oxygen. Okay, and then hypoxia and then glycolysis. How these hypoxia, that is if one that act on the glycolysis through the glycolytic enzymes and then GLUT1, and it acts on the LDH and it also acts on the PDK1. GLUT1, because of this, most of the glucose which enters into the cell, and then glycolytic enzymes like hexokinase, phosphofructokinase, these are all increased through the if1, and there is an increase in the conversion of glucose into pyruvate. But these pyruvate get blocked to enter into the TCA cycle through the activation of PDK1, and these pyruvate enters into the anaerobic glycolysis through the LDH. These orange color mentioned enzymes are all enzymes and then proteins are all activated by the if1. These are all the upstream molecules which stimulates the stabilization of the if1, and these are all the downstream molecules which are all upregulated and downregulated in case of hypoxic condition. Glioma, it is an intracranial tumor. It is one of the major tumor of the glial cells. Okay, the gliomas, which are the tumors that arises from the astrocytes or oligodendrocytes or from a ependymocyte. It comprises 30 to 40% of all the brain tumors, and it comprises 80% of the malignant brain tumors. The background of these study is based upon the glioma, which constitutes the common type of the primary brain tumor, and it is mainly characterized by severe hypoxia because this glioma is a solid tumor because the cells which are far apart from the blood vessels that are lacking the oxygen and results in the hypoxic stress. So as a solid tumor, this glioma is primarily characterized by the severe hypoxic condition. Okay, and then mechanism, that adaptive mechanism to this hypoxic stress or a potential treatment target, and however it is there is an increased understanding of these mechanism is critical for the development of an and implementation of hypoxia targeted treatment. So based upon this, the current study has investigated about the adaptive mechanism against this hypoxic stress in case of a solid tumor glioma, and how these cells that are metabolically reprogramming themselves and face the energy demand in case of even in case of an oxygen deprived condition. Okay, this is what the objective of the study. And then experimental design, when come to the study, when come to experimental design, even both the rat study and then human glioma samples both were undertaken in this study, and both their ethical clearance number is given here. And then in case of rat study, the rat is divided into two groups, that is group 1, which is uncontrolled rats, which is stereotactically injected with an DMEM medium that is supplemented with 10% FPS. This acts as a control rat. And then group 2, which are animals that are stereotactically injected with the cell suspension, that is a C6 glioma cells. Okay, 10 mole of DMM medium, which is supplemented with 10%, which containing 10 to the power 5 cells. Okay, under a controlled pressure, and these animals were housed for 30 days, and then the development of tumor was studied, and further the same tumor sample is used for further proceedings. And these observations from the rat model was extrapolated to the human samples for their validation. Okay, in addition to the level of glycolytic enzymes, the study also undertook about the metabolic variations that were studied between the grades of the glioma. And this is what the when come to the results section, this is for the histopathological uh, figure of the control and then group 2 rats. Even in case of a group 2 rats, you can find an uh, perinecrotic region with pseudopalliasing C cells and a condensed nucleus were seen. Whereas in case of control, we couldn't find any pathophysiological abnormalities. You could see an nuclear condensations and elongated cytoplasm in case of a group 2 that is implanted with the glioma cells. Then this figure B, which represents the tumor volume of control, and then experimental, that is an tumor developed glioma developed rats. You could see the tumor volume in case of group 2, and this tumor volume is calculated using the formula 4 by 3 into r cube into pi. This graph represents the tumor volume. And then first part is an gene expression analysis. What are all the genes that plays major role in the glycolytic aerobic glycolysis? The first one is an if one alpha. You could see the mRNA level of an if one alpha in case of control and experimental rat, that is group 1 and group 2 rat. You could find an increase in the expression of if 1 in case of group 2. And also this is what the immunoblot data of the if 1 of a rats, experimental rats. We could find an increase in the expression of if 1 alpha in case of an tumor developed rats. Similarly, we extrapolate this study in case of human samples too. Here, this G1, G2, G3, and G4 are nothing but the grade 1, 2, 3, and 4 of the glioma samples. We could find an increase in the expression of the if1 with increasing grades. That is especially in case of G3 and G4. Similarly, we could find an increase in the proton level of this if1 alpha in case of grade 3 and grade 4. There is a significant increase in the expression of this if1 in case of even in case of rat and also in case of an human sample. So there is an increase in the expression of if1 with increasing grades. This shows that there is an increase in the solid tumor volume. This results in the hypoxic stress, and which shows an increase in the expression of an if1 alpha. These are all the glycolytic enzymes that are all affected, that have an impact of the if1 alpha. These if1 alpha act on the transcription factor binding site present at the GLUT, that is a glucose transportase promoter side, and this plays a major role in the expression of these GLUT. We could find an increase in the expression of the glucose transporters like GLUT1 and 3, because these two are majorly expressed in case of a glioma. So the current study we have chosen these glucose transporters. We find an increase in the expression of an GLUT1 and 3 in case of an group 2 animals. Similarly, we find an increase in the GLUT1 and 3 proton level in case of group 2 animals when compared to control. When come to the glycolytic enzymes like exokinase 2, these exokinase 2 has an impact of an if1 alpha. These if1 alpha increases the expression of these exokinase 2 as it has an high affinity towards the glucose. So these HK2 get increased expression in case of an group 2 animals. And then pyruvate dehydrogenase kinase 1, we find an increase in the mRNA level and also the proton level of these PTK1 in case of a group 2 rats when compared to control. We find an significant increase in the expression of these glucose transporters and also in glycolytic enzymes in case of group 2 animals. This study is again overchecked with the human samples, we find an increase in the expression of the glucose transporters, that is 1 and 3 in case of high grades when compared to lower and control. And similarly, we find an increase in the expression of an exokinase 2 and then pyruvate dehydrogenase kinase 1 in case of an higher grades when compared to lower grades and also when compared to control. We find an increase in the expression of these proteins with increasing grades. As there is an increase in the tumor volume, there is an increase in the eye level hypoxic stress condition. So we find an increase in the expression of these glucose transporters and then glycolytic enzyme. And the next chapter is in chemometric analysis. In the previous study, we find a genetic analysis, genomic, that is a transcriptomic level of glucose transporters and then glycolytic enzymes. And then in the second chapter, we have seen about the chemometric analysis. Here we find an we use a human sample and determine the metabolite changes in case of lower and then higher grades. This is what the NMR data, stacked NMR spectrum of an aqueous metabolites from the human samples. This is what the NMR data of a control grade 1, 2, 3 and 4 of a glyma samples. We find an representative NMR spectrum that is mentioned with the metabolites with the help of a software chemonomics. Okay. And then next we go for principal component analysis of these data, NMR data. We given all the samples data and it is grouped into 5 groups, that is a control, grade 1, 2, 3 and 4. So this shows that there is some variation between the metabolites among the control and all the 4 groups. So it is divided into 4 groups. This is 5 groups, that is a control and then 4 different grades. When compared to this, this is what the 3D picture of the same. This is the 2D and then 3D image of the score plot of principal compound analysis. This is what the hierarchical clustering. These clustering which shows the this 1, 2 and 3, these samples are nothing but the control samples. It is clustered into one group, and then you can see the another cluster that is called lower grade. From sample 4 to 9 it is clustered, and this cluster act as an these clusters are the low grade samples. And then third group, that is nothing but an third cluster, it is nothing but an high grade clusters. We could find an uh, 3 clusters of the given samples, that is control, low grade and high grade. This study, this principal compound analysis, it gives only about the data, that is it says that the given sample is clustered into 3 types, but it doesn't give any details about the metabolites that are all plays major role in the grouping of these low grade and high grade. So to find in detail about the metabolites that are play major role in determining these 2 grades, we go for another study, that is nothing but an OPLS, that is nothing but an orthogonal projection latent square discriminant analysis. This In this analysis we have taken the group as a low grade and high grade. This image, this OPLS data gives the score plot. This score plot that clearly determines the 2 groups, that is a green color is a low grade glioma groups and this red color is an high grade glioma groups. So the given sample is clearly uh, distributed into 2 groups, that is low grade and high grade. So in this study we are going to find about the what are all the metabolites that are all plays major role in determining these 2 groups. Okay, here this is for the S plot of this OPLS data. The green colored circle and the red color circle the portion shows the metabolites that are all deviated between the 2 groups that plays major role in determining 2 groups. This green color shows the decreased metabolites, and then this red color shows circled metabolite shows the each bin is an each metabolite. This shows the metabolites that are all increased in case of an I grades. Next, in the next study of these OPLS data, we are going to find what are all the bins that is present in the encircled data. Bins are nothing but the metabolites that plays major role in the distribution of these 2 groups. In the next slide, we are going to find about the what are all the metabolites that plays major role in discriminating these 2 groups. Here we find an VPI, that is variable important in the projection score. Based upon this score, we are going to find the metabolites that are all, what are all the metabolites that are all increased in case of high grades, and what are all the metabolites that are increased in low grades. And this each bin that represents the top 15 metabolites that are all playing major role in discriminating these two groups are given in this table based upon the VAP score, that is nothing but a variable important projection score. If the score is greater than 1, it is considered as an absolute value for an correlation coefficient. So the VAP score that is greater than 1 are all considered for this group discrimination. So the top 15 metabolites that plays major role in discriminating these two groups are given in the table. You can see the chaurine that plays the top place. It is a high. It has an VAP scoring of more than 5, and we find this red color. Red color shows an increase, and then green color shows the decreasing in the level of metabolite. We say that in case of higher grade, that is grade 3 and grade 4, we find an increase in the level of the taurine. Okay, and then when come to another metabolites like creatine, we find a decrease in the level of creatine in case of an high grade. And lactate, these obviously there is an increase in the level of the lactate in case of high grade, because in case of hypoxic stress condition, the cell is majorly depend upon the lactate production for the energy uh, requirement. So obviously there is a decrease in the level of the lactate in case of high grade. And also glutamine, we say that the glutamine and glutamate also and another major energy producer in case of an metabolic reprogramming. We find an increase in the level of the glutamate and then glutamine. And interestingly, when come to the level of the L-myoinositol, that is decreased in case of high grades, because these myoinositol, this act as a major reservoir in case of brain cells. In brain cells, these myoinositols, these are abundant metabolite, and it plays major role in glial cells and functions as an osmolyte. Okay, whereas in case of high grades of glioma, we find a decrease in the level of these myoinositol. This is majorly because of the hypoxic condition. As a glioma, it is a solid tumor. It is more susceptible for an severe hypoxic condition. Because of this hypoxic condition, there is an upregulation in the level of the pallial pathway. Because of this pallial pathway, there is an increase in, obviously there is an increase in the level of sorbitol. This sorbitol, this competes with the internal myocytol level, stored myocytol level. So there is an increase in the polyal pathway with increasing hypoxic condition. There is an increase in the level of a sorbitol. This competes with the stored myoinositol, and there is an, obviously there is a decrease in the level of myoinositol. And then when going for a next slide, these are all the details of the metabolites, that is top 15 metabolites that plays major role in discriminating the two groups. Here we find an percentage average change between the low grade and high grade, and this is what the p-value and significance between the two grades. These are all the main metabolites that contributing the discrimination between the low grade and then high grades of glioma. This is for the overall metabolite pattern. From the current report, we have find that these are all these green colored metabolites. These are all the metabolites that decreased in case of an high grades, whereas these red colored metabolites, these are all the increased metabolites in case of an high grades. We already seen that there is an, obviously there is an increase in the level of the lactate, and the top first one is an taurine. This taurine level gets increased, and we find a decrease in the levels of myoinositol, choline, because the choline, this is for the major plasma membrane lipid. Because there is an uncontrolled proliferation, obviously there is an utilization of a choline for a synthesis of a plasma membrane for the new cell. So obviously there is a decrease in the levels of this choline. So these are all the metabolite pattern that we find in case of an glioma. And then the figure B, this shows the top 5 perturbed metabolic pathways. These are all the top 5 disturbed metabolic pathways in case of low and high grades of glioma. The first one is alkaline metabolism. This is what the topmost metabolism that get disturbed in case of an high grade and low grade. And then glutamate and glutamate metabolism. These are all the cake pathways, metabolic assay. This is what we use a software called a metabo-analyst. Through this analyst only, we have carried out the principal compound analysis, OPLS analysis, and also this pathway analysis. Through this pathway analysis, we get a top 5 disturbed pathways, that is a choline metabolism, glutamate and then glutamine and metabolism, glutathione metabolism, charin and hypotaurine metabolism, phenylalanine and then tyrosine metabolism. These are all the disturbed metabolic pathways. So, the summary of the current study is we found that there is an increase in the level of IF1 in case of an rat when compared to control, that is glioma induced rat when compared to controlled rat. And also we find an increase in the IF1 level with increasing grades of glioma. Simultaneously, there is an increase in the glucose transportase level in case of glioma-induced rats, and also in case of an human glioma, that is a high, especially in case of an high grades of glioma. And also there is an increase in the glycolytic enzyme level, that is a exokinase level, and then PDK level were studied in the current report. And we find an increase in the level of these two enzymes in glioma-induced rats and also in case of an human glioma samples. So from the chemometric analysis, we have identified 15 characteristic metabolites that plays major role in discriminating low grades and then high grades. Okay. And also we find and 5 top disturbed pathways that all plays major role in discriminating these two grades. So from the current report, we have find that how these if one that act on the glycolytic enzymes and also how it act on the metacellular energetic pathway to adopt the cells even in the condition of an high energy demand with low oxygen level. So the if one that act as a major role by increasing the glycolytic enzymes, and it increases the glucose transporter levels. By through this, the is if one plays major role in adaptation of the tumor cells towards the hypoxic stress. And the conclusion from the current study is, together with the gene expression analysis and also comprehensive metabolomic analysis, the current study defined the adaptive pathways of hypoxia in case of cancer cells, and also from that it is concluded that hypoxia induced metabolic alteration plays a major role in adaptation of cancer cells towards the survival even in the condition of a low levels of oxygen. So these new insights may be further extrapolated for the development of treatment strategies against hypoxic and then highly aggressive tumors, especially in case of glioma, with the natural drugs to overcome the side effects. And then future perspective of that study is glioma, not only a glioma, even the cancer as a sweet tooth, because it utilizes the high amount of the glucose. So, when compared to that of a normal cell, these cancer cell which utilizes more glucose because it depends upon glycolysis for energy production. In normoxic condition, a single mole of a glucose produces 34 moles of an ATP, whereas in case of anaerobic glycolysis, this glycolysis produces only 2 moles of ATP. So it depends upon more glucose intake. So this cancer cells, it is the term which express tell about these is cancer which are having a sweet tooth. So to overcome this, we can use any analog, that is a glucose analog that can prevent the anaerobic glycolysis, can be used as used for treating this metabolic reprogramming and adaptation. And also second aspect is nothing but an metabolic symbiosis. This figure shows that the top this is what the tumor here, the blood vessel is present here. These are all the aerobic tumor cells because it gets an oxygen from these blood vessels, whereas these are all poorly oxygenated tumor cells. So these cells which is under the hypoxic stress, and these cells the metabolic reprogram that is carried out in the cell hypoxic tumor cell have an impact on the aerobic tumor cell also, because the hypoxic tumor cell which highly depend upon the lactate production for an energy demand. Okay, so the lactate, high levels of the because these pyruvate can't enter into the mitochondria because of an increased level of PDK1, which blocks the entry of a pyruvate into the TCA cycle. and these pyruvate is converted into lactate because so these cells are highly richly concentrated in lactate. Even in an chemometric analysis also we find there is an increased amount of the lactate in case of an high grades. So these higher increased lactate have an impact in the aerobic tumor cell also. These cells which get an oxygen from this blood vessel, but it rather than using glucose for an energy demand, these cells take an lactate that are released from this hypoxic cell. Hypoxic tumor cell, this is an aerobic tumor cell. So these aerobic tumor cells update this lactate that is through the MCT1, and these lactate enters into the aerobic tumor cells, and there is an conversion of these lactate into pyruvate, and this again enters into the TCA cycle. Because these cells are aerobic tumor cells, it is having an sufficient amount of oxygen to oxidize this glucose into carbon dioxide and ATP plus water. These take these lactate and convert it into a pyruvate, and these pyruvate enters the TCA cycle. This is what the symbiotic relation between these two cells, that is an hypoxic tumor cell and then aerobic tumor cells. So these are all symbiotically related. How these lactate from these cells is utilized by the aerobic cells, and these cells, as it utilizes the lactate in higher amount, the intake of the glucose by these cells get decreased. So these glucose are available for this hypoxic tumor cells, because these it is totally depend upon the glycolytic ATP production. So it need an higher amount of glucose when compared to normal cells. So these cells take this lactate, and then the glucose, that is excess glucose, is taken by the cell. So they both are symbiotically metabolically related to each other. It supplies lactate to this aerobic cell, and then the glucose that it is not taken up by this aerobic cell is utilized by this hypoxic cell. So they both are symbiotically related, and this glucose is taken by these cells. So the drug which act upon this MCT1 and MCT4, MCT1 plays a major role. So the future perspective of the study is any analog, glucose analog that can be taken used to prevent the used to stop to control this anaerobic glycolysis, and also we can stop this metabolic symbiotic relationship between an aerobic and an anaerobic tumor cells by blocking this MCT1. This is what the future perspective of the current study. Thank you. And then I would like to acknowledge my mentor, Dr. AJ Vanistry, who is providing me this opportunity. And I would like to thank UGC BSR for providing fund for this work. And I would like to thank SNCI and Ethiraj College for providing me this platform. At last, I would like to extend my thanks to Department of Biochemistry, University of Madras, Guindy campus. Thank you. Thank you, one and all.
Thank you, Mom, for illuminating us on the advanced studies based on the metabolic programming in glioma, and thank you for telling us about the future perspectives.
Thank you.
Undeniably, this session was totally captivating. Thank you once again, Mom, for joining us.
Thank you.
With this, we come to the end of our technical lectures. The commencement of the offline events will begin shortly. I request all the dignitaries, staffs, and our incredible audience to join us for the finale, which will commence by 3 p.m. Thank you, one and all. A very happy afternoon to one and all present here. It gives me great honor in welcoming the dignitaries, staffs, and students to the finale of NEUGLIA 21 Regional Virtual Symposium. I now request Dr. Safia, Associate Professor, Department of Biochemistry, Ethraj College for Women, to take us through the finale.
Good evening, all. Dr. A.J. Vanishri, professor in the Department of Biochemistry, University of Madras, Guindy campus, is engaged in neurobiological field with specialization in molecular aspects of neurological disease, probing solutions for the unanswered complications. Identification of novel key molecular signatures in critical neurological disease such as glioma, schizophrenia, and Parkinson's disease are made by her laboratory to prove the significant contribution to the arena. Recently, the first time report was made on myelin and its components, a completely unexplored pathology in the stress and stress-related disorders, which is considered as very significant. She has secured research grants from prestigious national research funding agencies such as DST, ICMR, and also WNRF, highlighting the significance of research arena. Her work are published in many peer-reviewed national and international journals with good impact factor. Presentation of her research outcomes in many national and international conferences by her research students and by her has won best and first prizes. Her laboratory has filed 2 patents that are anticipated to contribute to the welfare of the participants with neurological complications. Further, the lab has also probed and reported the possible mechanistic aspects of traditional drugs against neurological diseases using various types of models with successfully standardizing methods for development of alternative models to rodents, which is crucial for research in neurological disease. She is a lifetime member of various social societies, including International Society of Neurochemistry, Society for Neurochemistry, SNCI, IABMS, etc. She is the regional coordinator and has initiated Chennai chapter of SNCI in the city and has organized academic meeting representing the chapter. She is a reviewer for many reputed journals, national and international journals. She has served as member of various administrative committees. Ma'am is a volunteer in non-government organization that provides service to the society. She has also completed certificate course on healing models based on traditional knowledge. It was Dr. Vanishri who seeded the very idea of this symposium and nurtured it to bloom today. May I now invite ma'am to deliver the finale note. Thank you. Ma'am, you're on mute, ma'am. Excuse me, ma'am.
Yes.
Yes, ma'am.
Okay. Shri Matre Namaha. First, at the outset, I would like to bow my head to the divine universal power which is directing all of us. I happily congratulate and appreciate, of course, very with immense gratitude. I appreciate management of Ethraj College and Department of Biochemistry under the headship of Dr. Sujata, who had taken up this event and they were willing to come forward to have a joint venture with University of Madras, Department of Biochemistry, and they are also joining our hands with our chapter, National Society for the chapter. I'm not audible. Am I audible? Audible? It's on. So, I wholeheartedly thank our presidents of National Society, Dr. Thakur sir, and Dr. Prakash Babu sir, General Secretary of SNCA Chennai, Vice President Madam Rita Christopher Madam, who are all driving the force, the neurobiological field to be nurtured in our country. And I immensely, I owe to tell my thank and gratitude to our HOD, Dr. Limon Valechano, whom you would have met yesterday's meet. And also, I'm very much finding no words to thank our former HOD, Dr. S. Niranjali Deviraj, who is still the backbone of our Department of Biochemistry Gindi campus and also still driving our directing and helping to run the department even now. Other chapter members and my friends for Society for Neurochemistry and other regional coordinators, all members, they are all playing immense role in nurturing this event, and they are very highly supportive and making us to enter into this kind of a task. Yeah, I first of all, as you people have seen in the brochure, the participants, the faculties, to all people whom we have circulated by this, very eminent college personalities, you would have seen the objective of that particular this particular event, nurturing the neuroscience and bringing up various proceedings, basics, even basics. That is the title you would have seen towards this. Uh, Towards this event. So I would like to tell some important points on the crux point of the event. The crux point of the event, where we are talking much about stressing. On for the past 2 days, you have been witnessing some developments on the basics and how to manage this and that, and stress and various related disorders that might crop up due to the stress, etc. And this era is again more. I feel that if you take a statistics and you go for some Guinness record, this word is being uttered for much more times when compared to other this thing. I thought that might be an important area to touch upon, and that too whenever I meet after this lockdown or some leisure, when we are somewhat experiencing some changes in our daily life or routine. Even the students, they whenever they meet in person, they are all concerned about the stress they have undergone in the past few months. Of course, a year, more than a year, or almost twice, 2 years. So we have to know what actually this stress, what will be the biological background of it, and is that essential for us to maintain some uh, how you have to maintain. You cannot say maintain and manage, but you have to think how to go about it, colloquially speaking. We have to learn certain things accordingly, because man is evolving, continuously evolving, and we have to go with the stream. So, what I thought of giving as an input today, I might be as a complete. This is not a valedictory address, but I just wanted to conclude this 2 days event successfully, run by our joint ventures from two departments of biochemistry from Ethiroj College and our own university and Chennai chapter. So, how we are going to make an impact? That's the only what to say. That might be the main aim. We have to compile what we have seen. The compilation will be done by our counterparts, but we'll try to see how to manage or think with some technical inputs. That will be responsible as a regional coordinator. That's what I mean. So I thought I'll share the my some of my insights, which I feel that might be useful for the younger generation. It is happy moment for me to share that kind of my insights when I want to uh, if I want me to deliver some note on this event as the concluding remarks of this local chapter meeting. Okay, let me share the screen. If it is visible, please visible, you can tell me. Yes. So I'm sharing the screen. You people are trying to see what is there in the screen. And the question is like this, though we are trying to frame the thing like, do we, basic insights, we have been discussing basic insights against neurological stress. Do we really need to act against stress? That is the question that you should ask yourself, whether you need to have to fight something against, you have to see that as a big deal and fight it. So I feel that the answer, if you see that, definitely not necessary, at least not always, because stress is a natural process. Just a moment, please. Yes. Just a moment. If I'm audible. I am trying to.
Yes, ma'am,
Yes,
you're audible, ma'am.
this is visible. Very good. Thank you. So I don't want to repeat that. The question is like this. I hope you would have heard me telling that you need to ask the question to yourself whether we need to really act against the stress. And I feel that you need not you need not worry about asking I mean, what to say, that it is not always because it is a very natural event that can occur to any organism. That simply is an adaptation you body or your mind is experiencing. So you need not fight against that. It is not that essential. And this type of categorization many of you might be aware of, that is whether it is an eustress or the distress. People would have heard about these two terminologies. We don't want to go into these details because this is psychological framing of answer to this or definition for this, and other are medical framing and normal layman's framing. Everything might be different. This is Greek and Latin, you and this stress, Greek and Latin, and it is not that important for us. But I want brain will always learn from the known to unknown. So that is the basic point, which why I'm trying to show this. From this point of already known information, let us sail through some more aspects which some of you might be already aware of, and for younger audience at least that might be helpful. So all these things which are a few minutes coming, upcoming minutes will be just occupied by some of my inputs based on the experiences, and also but our own experience or the people's experience, and also based on the experiments what we have done in our laboratory. So that kind of data, because the always the younger generation will be asking for the proof, how you say this advice or how you say this notion, how do you say all these things without the proof. So that is why I took, because some are elder people, yeah, they'll be trying to tell us something based on their experiences. I might also tell something based on my own experience, but it is all in your hand that you should be able to grasp things, provided you are very That is based on whether you are a person who might require proofs or evidences before you could understand or believe all those things. Anyway, that's the thing. That's why I'm telling you some points I might be delivering here based on the experiences and also based on the experiments. And the structures are all shown here. The brains, right from earthworm and Drosophila or chimpanzee, we are not using some, rodent models we are using, isn't it? All those experiments, all what we have seen in the day, because this is not a technical session. Please don't worry. I'm not going to again make you to pressurize yourself, because you have been hearing much about the other speakers of yesterday's event and then today's session as well. Not the technical details, just ease yourself, and just we are going to sail smoothly with some evidences. That's nothing to worry about it. And you just hear like a story, and then if you people are trying to listen to a story, how will you feel? That sort of this thing only, because some of the heavy points might also be put forth, which might be understood only by the elders. Okay, so let me try to compile this based on experience. What we thought, I myself thought like before, without. Going for the conventional mode, I thought it could be simply put like this: exogenous or endogenous. Exogenous, extremely people would have talked about it. You also know what other people or other external factors or other environmental stimuli, all those things are going to be having, may have some impact on you. And I'm not going to detail on this various issues like interpersonal skills, communication, time. Yesterday also, Dr. Bhatia has clearly, lucidly presented how you could go about it. The source can be the mobile phone usage, but various suggestions he was telling you, and it is also widely supplied by many sources, e-sources or the elders. You should spend like this, but just leave it. Endogenous is, what do you mean by the endogenous? Because you might be wondering why this kind of categorization man is trying to put, instead of eustress and distress, which is widely globally followed by many researchers or biologists or psychologists. This endogenous is what you do. No, you people are all life science students, and I'm sure that what you know about the terms like pharmacodynamics and the pharmacokinetics, what the body is doing or the drug is doing to the body. Likewise, the endogenous is something what you do to yourself, be it a variety of stressful events or the people around you, but what you are going to, you intrinsic, it's quite intrinsic. Endogenous is quite intrinsic, irrespective of the stressful conditions. Irrespective of the magnitude of the stressful condition, how you are going to view it. You got it, view it. Endogenous, that is what I call it as an intro. So it is purely based on you. I don't bother about the amplitude or intensity or the number of the stressful events, but how you are going to view it, or your own perspective or the intrinsic view, we call it as endogenous type. And for that you need to have an idea, like all these are might be guidelines if at all really you would like to come out of the stress. And you have to first of all you recognize, which you label with your conscious mind. Consciousness is something very important term that may have very good meaning, which we are not going to into the details. But as of now, the word consciousness has got some understanding. You might be having some understanding on that. So consciousness, here consciously, how you recognize. First, you have to recognize whether this is a stressful event or the real stress, or this person is a person who can make a stressful situation, bring about a stressful. So first of all, recognize it. And then you have to ask the question like, is that essential to give attention to that particular stressful event? What do you mean by that? You can ask again two more this thing. Attention if not required, what should be done? Attention if required, what should be done? That you need to analyze. Then you can take some actions. Actions generally people all know is medical intervention or advice or a psychological. Paramedical, I just simply talk about the clinical nutrition or some exercise or the yoga, or non-medical will be like that, natural will be like that, and some people will go for the spiritual mode of alleviating their stress. These are all certain guidelines that a person can think about, apart from the important advice, whatever already you have been given by various media or elders, or things like that, like no alcohol, no smoking, or diet, or this and that. So these are the questions that you should frame in your mind and find out some answers, like recognition, attention, action. That's it. Nothing. I'm just putting that in a simple word. Then if you come across each and every stage of this type of thing, then you will come to know. Finally, sometimes you may also end up with such a conclusion. It's not at all a stress. It is a simple an activity. Where then how to go about it? You may have several ways. These are all personalized. So it's a guideline I'm trying to say. And what I would like to say is, all these things, your attitude, your basic character, your habits, your behavior, and your understanding level of the philosophical understanding, to give some idea, I have put that word, but it also have some deeper meaning. You have how what at which stage are you in, and what sort of an understanding you do have about life. Okay, about life, and what to say, if you believe in God, if you don't believe in God, if you believe in nature and you don't believe in nature, and if you believe in rebirths, everything may also count. All these are based on your beliefs. Beliefs may be of different kinds: false beliefs, true beliefs, real beliefs. Yeah, and so these beliefs are going to several factors you can put as a subcategories of all these, majorly in whatever predominantly listed out here. So the attitude and character is also prominently playing an important, very, very important role, that may have some impact on your well-being, because you will need to live a good life. Yes, so that's why you are telling something. You have a routine in your mind, and sometimes you feel that that routine is being broken up, and then that is due to this type of stress, and this type of stress is due to this. So naturally, they are all interlinked. We are entangled with such kind of this thing. So they may have, these people have to understand. I mean, we have to understand that these factors of your own. I'm not talking about the external. I'm not talking about an external. So you have to, not clear? பண்ணிட்டேனே எதுல இருக்கு? So I need to, whether I'm audible, and then I don't know why it is. It is show that starts sharing only. It is sharing only. Hmm? Is that okay? Yeah, it should be seen. It should be seen.
Mom, we can see your slides now, mom.
Yeah, second slide was that visible? Definitely not. And is that visible? Yeah.
Yes, mom. Yes, mom. Mom, we
Third
can see.
slide is also visible.
Yes, mom. We can see, mom.
Yeah. Then I told you we are talking about the experiences and the, this visible.
Yes, mom. It's visible,
Yeah.
mom.
Very good. Then that's why I was roaming around this point as well. This also I told you the influential factors. That's where we are in right now. Yes, this visible. Oh, fine. Shall I go
Yes,
to the
ma'am.
next? Very good, very good. So this is yeah, this is very important thing that me people should be able to follow our own self. That's the more important rather than seeking other. Okay, that's we will conclude in. We will put that as a simple one in last slide. So what I'm thought this is just based on the views and our own understanding. We are listing out certain things. Okay, if you are able to understand the influential factors of, you can always bring some changes in your attitude, some changes in your own character, some changes in your habits or the behavior. Of course, the stress is going to decide the behavior, right? So you have got it. You got the point of the philosophical understanding as well, I think. So now I thought of showing our own technical data for a couple of minutes. Then I'll conclude as well. My talk will not be that lengthy. Yeah, we have used several, as I've been displaying the various brains of small animals, right from earthworm or the Drosophila or rodents, and, even the humans. Okay, so we have made some small findings based in what is possible in our laboratory. So I thought of providing this detail to you. So this is the interesting because rat model is very what to say, very familiar to many people here. So even the PG students would have got some MSc projects outside, or in your own department you would have used rat models. As I told you, I'm not going to talk about the technical details. So we have used rat models, and we have subjected those rat models to the stress conditions. That is, experimentally, we have to induce stressful conditions to the rat models, and what happens, and also we have to see various modes how to mitigate such kind of stressful conditions, this and that. So that was the experimental setup. You have various stressors that can be useful for creating an experimental state that can simulate the condition of the human being who is under stress. One of the method which we have used is the immobilization stress, chronic immobilization stress. So after subjecting that, the colorful pictures on the top and the wheel-like pictures on the top, we don't get bewildered. They are the behavioral pattern. We was mentioning about the behavior as well, isn't it? So behavioral pattern that might be affected in these stressed rats, and what exactly the biochemical and the molecular changes, the gene level changes. Our job is to do all these things. We have been checking for all variations. Yeah, it is very what to say, disappointing to note that stressful conditions can have various effect at your gene level. Everything manifestation is your behavior, and at the cellular level and gene level, yeah, changes are there. That is worrisome, but we need not worry about the worrisome points, isn't it? We should also think about the ways to come out of it, and think over it what should be done. So that also we have done to our rats, and we have exposed that rats models to special conditions. It is not so special, all technical. If at all any people who are involved in this research, they might be easily able to understand. And for the novice people, I could say that you call that as an enhanced environment. That enhances, the word itself denoting something to you, isn't it? You are improving the environment or environment, whatever way you pronounce it. Then you find it like the place, the exposure of such kind of environment to the particular rodent model showed tremendous improvement in their all this. We like this is just a maze we have used, and you can always expect beautiful and encouraging findings that can be presented by the rodent models when we are exposing them to different environment, from even though they are already stressed, so we have to check at various levels. We have seen some changes at the cells. We have done our culture studies wherein you can see the cartoon over there, right? You can see the cartoon over there where I'm trying to show some of the neurite like thing. We have seen how this stressed condition affecting the neurite lengths, small outgrowth of the cells which we culture in a dish, and how any there is a difference in the length of that particular red color. That at least if you notice, no, that will be more than enough. Don't worry about other scientific inputs. So I thought I'll make this note as a story one, like not a technical. So you have to find out the red color length. They are different, isn't it, in the cartoon? That's what also shown by our results. So when we have checked we had softwares and we had done our we have exposed the condition rats to the different conditions and sacrificed, and hippocampal region was used for various analysis. And one such thing you can also find out how you can simply regulate that, because yesterday Dr. Prakash Babu sir was also mentioning that we are all still looking at how the brains are looking, how brains are working. So the problem here is we do have some misunderstanding about our own brain. That is the situation right now. We have understanding of our own brain. Of course we have all, we people say that you have misunderstood me, but we have misunderstood our own brain. The brain is a wonderful instrument. It is not your part actually. There is, if you underline this word, if it is not a part of your own body, I have a philosophical orientation, I don't want to touch upon that. It is not part of your body. But of course, if you are not looking at the physiological point of view, and if you are looking at your own physiological point of view, you can see that it's a best instrument, and we should not we should not be misunderstanding this wonderful organ. And there were some concepts in the earlier yesteryears where we were thinking brain is like this, it cannot do this, it can do this, likewise. Then some years later we no brain like this, you have not all the cells are not terminally differentiated, you have neurogenesis. And then some people say neurogenesis possible here and there. Some immature scientists told that. And sometimes they are also trying to say that no, you have a very 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 small population of the cells where you can expect some neurogenesis. This, so of course we are able to find some regions of neurogenesis, what you call it as an adult neurogenesis. That is the only very very happy message that I'm trying to put across the younger on generation. Your brain can do whatever you want. So because they are also, unlike the initial notions or the initial concepts, you are able to rewire. Got it? You are able to rewire your brains. So that's what our data are trying to show. And not only that, as I was mentioning, we have used zebra fishes, and some of the behaviors of the zebra fishes. We have used how they can prefer, how can they move, prefer colors, right? How can they move this and that, and etc. All these things we have done. Interestingly, for the first time in the literature, at least now I think we have some more results. At least during the initial stages of our study, we are the first to show some interesting changes in our myelination pattern. No, all you people would have studied, even quizzes would have completed, you would have got some preparation, and some people would have got some good marks in that. Myelin, axon, So I'm just taking that simple examples. So we have done some research in that part. Surprisingly, we came out some important findings. Here you can see how these myelinated neurons are there, what lipids might be there, and how can the electronic macroscopic picture is also showing the structures and also the cells that are part and parcel of this, the myelin, the oligodendrocyte, the membranes, major lipids. So we have done some research on that and collected some data showing very very encouraging note on the myelination as well as the lipid components, which are important for various types of functions that should be performed by a brain. And this also is giving us a good because forebrain region, very very important for us human being. At least, there are difference in the brain types and the hemisphere, this and that. What rat can do, we cannot do. What we can do, rat cannot do. In spite of that, of course, we need to use some animal as our models, and we are using that. And we do have a well developed prefrontal cortex and forebrain, etc., which is going to be performing various functions unlike the rodents. But in spite of that, we are using forebrain of these rodents and find out what sort of change that might occur to them. That is important for you. This Your forebrain is all about the important faculties of the mind. So that forebrain is also, and the myelin regions in that, when we examine, that was giving us, we have used some Lexel fast blue staining, and they are giving us some importance of this lining of white matter, and the architecture of this white matter, how do they exist in the stressed condition. And even under the stressed condition, if you manage to provide some good exposure to the animal, what sort of positive changes that can be brought about in the brain. Yes, that's the idea. You are able to rewire if you are exposed to a good environment, or if you, as I told you, if you concentrate more on the endogenous, because you should not complain me at the end of the talk. You are talking about the exposure, we are not exposed, but it is, that's why I'm just instigating on that list which I have shown in the slide, in the previous slide. Like, endogenous factors are highly important rather than the exogenous. For rodents, how can you go for the endogenous factors? So we are talking about the extrinsic environment here. Okay, this is I'm also trying to give you the students who might be curious in asking several questions. I didn't want only to give the behavior. I didn't want only to give the cellular structure. I also want to give you the proof ultrastructural level, at the ultrastructural level, because at the ultrastructural level, what you see here, everybody is aware of transmission electron microscopy. Is would have studied the aeronautical paper. That ultrastructural appearance of this myelin is also presented for your benefit. Don't worry. If you are going to if you are able to come forward for not the fighting, I'm managing all these data words I'm not going to use. If you are going to take care of yourself, that self-care, that doesn't mean that you should disturb others. Okay, that self-care is beyond something that if you are going to take care of yourself, you are every level, at the cellular level, at the gene level, at the behavior level, at the microscopic level or macroscopic level, you will be bringing about a lot of positive changes in you. So COVID and all, nothing. You can always do your own. You can take care of your own activities and other what to say perspectives to, if at all some modifications or revision is required, you do that. Will try to provide lot of that will fetch a lot of benefits to your brain health. So finally, you are going to do best in your life or your exams or whatever it is. Yeah, this is the detail. I don't want again go for the technical, as I told you. And for some more research oriented students, I thought not only this macro or micro or this thing, you can always also look into the expressions of very important markers of the myelins, because myelin and the stress and the markers we have reported for the first time, and how they can have changes in the myelin basic protein and oligodendrocyte protein, glycoprotein. All these things we have checked the expression in these animals, and then we are successfully showing how even a stressed condition can bring some detrimental effects to these proteins or the markers or the myelin or axon or the transmission. How can you bring it some, you can have some normalcy, provided you change that particular environment. Okay, so not only that, I have to also show you a proof from our own species, isn't it? If I might go on with some evidences from zebrafish or the drosophila or rodents like that. But I need to also show one of the evidence which might be helpful for you to have the strong point in your mind that it may have effect like this, and if I change myself we can have the better life. So we went on to human models as well, like we had selected some population, and we have seen some changes. Only one report I'm trying to show here, because this is again here, first finding, it is not reported earlier, or most of the findings which I am trying to share for the proofs to show what you need to do in your life, maybe in the moment from now onwards. I'm trying to show this, only one such kind I'm showing. Again, I'm the person whom yeah, the importance should be given to modern equipments, methodologies, etc. Also, I'm the person who can be, I mean, who believes even a small idea or a small instrumental setup can have much impact on the science field. So in that regard, we have simply used some flow cytometry technique and lymphocytes to find some changes, what may happen in the blood sample, what kind of an indicator. I cannot say that as a biomarker, it's a big terminology, you cannot use it here. So what sort of change that a person can exhibit when he's under stress or when she is under stress. One clipping from such kind of study, after the obtaining proper ethical clearance, what we have seen is a factor, pituitary adenyl cyclase protein, which is going to have an effect on, you know very well, HPA axis. HPA is playing an important role in your endocrinology. So we have selected this protein, and then that is showing some interesting changes. You can see the scattering. Many flow cytometry, I mean, people might be understanding the technique over there, following the scattering principles. We have used the samples, and then we have seen the staining pattern where you can see there is a complete when we subjected the data to this software, you can see express the results as a mean in fluorescence intensity, wherein we are able to see bigger change comparatively. Alterations in these two cases of control and the stressed people. And here this type of finding might have many implications, and what might be happening at the body level and what should be done, those things next these all coming next and next. Now this type of small indicators in your own sample will be helpful for you to have an idea about the condition you're undergoing, and immediately what could be done. And you could just do that and at least go proceed with your own life with maybe after implicating or incorporating some important changes in your life, which might be helpful for you to avoid. So I'm not continuing with the data. I'm just stopping here. So if you are able to ask such questions, is it possible to live with stress? If you ask me, yes. Any stage of pressure is manageable? Yes. At any age, it is also yes. Right from, because now when we are all in our school days or when during our childhood, we never used words such as like stress, because our Indian country and the family setups are like that. It may come, it may arise, it may go off, it may fade off. Everybody might be on what to say, agreeing with me. I don't know, people are giving more stress on to this stress. You should not, first of all. And every child of at home, maybe they are single child or whatever it is, people are trying to say they are stressful, and parents are also trying to promote this kind of a feel in their kids, telling that no, my student, my son or girl or boy are stressed. Why you need to do that kind of promoting way of I have to have a approach like this, promoting the condition. That's why I'm telling you, we during our childhood days, we never complained of stress. You cannot say that no, that age is different. Every age, yeah, you are evolving, you you have technical changes, you are technological changes, you are evolution is there, but you please evolve in a positive context alone. Never complain that unlike your days we are exposed to this, your gadgets are like that. Anyway, even now elders are using the gadgets without getting stressed. The mind makeup or the perspective on how you manage and how you had, because one of the slide about Dr. Bhatia sir, if I take that, you could have seen that the connectivity, family connectivity, it could be a spouse, it could be a brother, it could be a sister, etc. That is anyway you're a human being, and years to come also we are going to be in the same species, homo sapiens, and you need to have a proper bondage which is not found today's world. That will helpful for you to overcome. You have to have a good, that's why I listed all those things: your own attitude, your own behavior, your own connectivity, all those things, because that's sort of an perspective like how you view the current situation is stressful, or that you please avoid that. It is possible to live with stress. First of all, that you need to mind, I'm stressful, life is like that. We don't be complaining that you, whatever tremendous pressure you are experiencing because of the COVID or because of the online education, because of the teachers, or it's for students, and again the same thing in a different manner, same points in a different manner for teachers. You know how they are suffering, online teachers especially. Your madam, and they have the this switching over of offline to online, how they are suffering to make you to be in connection with their students. So, but anyway, it is one important thing you should keep in mind is this is again based on some of the students who are all coming to me and telling that it is highly stressful and how can manage, and some more, some unpleasant tendencies they have, all those things. That's why I'm telling you, let it be there, let it be there. Stressful condition, you can still live with the stress as if you don't have any stress. So that is one thing you need to put in on this point. This point will create a neural life firing network in your mind. Please take my point. So yes, it is possible to live with the stress. This point or this thought, this deep thought, definitely will ignite a neuron in your mind. Got my point? So any stage of pressure is manageable, yes. Any age, and also it got no age difference. Any age you could, maybe you're a teenager or toddler, of course, yet to come. A toddler might also come up with I'm stressful. Okay, leave it. Other important thing to conclude before I'm just nearing my concluding remarks. I'm trying to, after inculcating this point in your mind, that you say S for your stress. Please say S for your stress. And how to go about it, you can see in the next two slides. If at all it is able to ignite something in you, I'll be very happy. I'll take only one influential character. Is what you mean by that? Habits. Yes, and other factors let us leave. So one influential factor is, if you remember my slide, you can see that habits. This is easy, no, for you to your daily routine, your habit, your reading habit, your eating habit, all those things. Apart from this, you might be telling that drinking coffee or drinking tea, painting, that might be a hobby or a habit. Okay, if you are having all such kind of any people, cannot be. He will be with some habit or the other. He cannot be found as devoid of any habits. So what you need to do is, you just take this happiness and joy, irrespective of the condition. Happiness and joy should be made your habit. That's it. You may have different habits, but you take this happiness and joy should become your habit. So how to do that? If you want to bring about changes in you, that you should not be worried, you just have to do this. Yeah, இந்த முள்ள முள்ளால் எடுக்கணுன்ற மாதிரி அந்த ஒரு stressful habit you have to take, like, you bring this habit into your mind. Joy is my habit, happiness is my habit, because philosophically speaking, you are a person or you're not a person, and an entity in the universe. with innate happiness. That is the nature. You got it? So you are the entity of the universe with innate happiness in you. So we are what we are doing, we are forgetting that. That's the thing. We are forgetting, we are at least made to forget that we are an universal part or entity of the universe with innate personality or the habit like this happiness. And so you try to what to say, at least try to nurture that type of a hidden talent in you. Got it? So how you could do that? So all these things are little bit hard working. I mean, they may seek some hard work, they demand some hard work, but you need to do that provided you want to come or live with the stress happily. Yeah. So irrespective of the exogenous condition, endogenously you seed all these things, intrinsically you seed all these things, that is calmness, stable and inert. Again, this is where young minds may not be able to capture the inner details of it, but of course experienced fellows or your teachers and your adult people, they can immediately grasp the background meaning of it. It is again a heavy subject. If I take each and every word mentioned here, I need to talk something on that, but we will keep it short. And intrinsically the calmness, intrinsically the stable state, intrinsically the inert state, it will all helpful to what to say, be helpful for you to cultivate that habits. Cultivate what habit? These two, happiness and the joy. Feel the feel, and that is again, I'm giving you the protocol. How could you finally arrive at the result? For these calm or the stability of the mentioned here, one more part should be there, what you call it as a detachment. Detachment again, it may have different meaning or different context based on your understanding. That's more important. Based on your level of understanding, whatever I speak here might be differing, but there is only one meaning which will be helpful for you. But I give you the freedom based on your level of growth or maturity. You may have some understanding. Okay, so the protocol is anyway like this. What stage are you in? What age are you in? You are with not living with stress or high stress or low stress. This is the protocol. This word should be there in your body and mind, and that might be helpful to cultivate the habit. And that habit will definitely help the coming generation or future generation, or I mean the people here to have make this world to a better place to live. Finally, that is what we need, isn't it? Make the world to better place to live in. So the detachment is you have to show that also be seeded very strongly as an intrinsic quality that will help you to grow or evolve in whichever way you decide what you want to become, and whichever you decide how to live. When you still rise up early in the morning from the bed, you decide which way the day should go, irrespective of whatever situation or people you meet or situation that may be experienced by you on the whole day. But how to go about it, the last word might be helpful. And that sort of as I told you, the understanding might be differing, and you can see how you can manage or understand the detachment. The English meaning or sometimes straightforward, sometimes they may have several hidden meanings. Sometimes if you have to refer some books and this thing, you can take this word in order to have your own understanding. You can see that whatever road path is paved there or footpath is paved there, we have to walk with our own footsteps. So the road map only I'm giving you. You got it? The road map or the protocol only I can give you. The footsteps is by your own self. So I think that may be useful for you. Anyway, I'm trying to make it still more less complicated. Of course, stress, it is initiated by the letter S. So what you need to do, think about think about three more S here: stratify, spark, and spirituality. Spirituality I'll come little later. Don't get you will not worry about why the word spirituality should always come here. No, spirituality is something different. I'll come to that later, little later, next in a minute. Okay, so stratify. So one stratify the mean that means, as I've written there, you know, you decide. I told you intrinsic features as recognize, etc., isn't it? And then whether attention is required, you need to give an attention, you need not give. So that's what I'm here putting as a summary here. You decide whether I need to work with it, or whether I need to work through it, or whether I need to live it. So all these things will be helpful for you to think about the situation and take the steps. Carefully follow. I'm not using the word against. Let it be there. It's a natural process. Of course, it should not be complicating the life. That is there. So your attention is needed. So when you have a situation, you ask these questions to you. I need to, first of all, you decide the objective in you. I need to stratify. How to stratify? To work with it, to work through it, to live it. That is up to you. Okay, so these things will help you, and definitely that might helpful at least to cross the at least half. Then you are done halfway. Okay, you can cross that tangles of stress, if at all you label that it as a tangles of stress you got in the middle, etc. If you can do something about it, do it. Okay, if you cannot do it, you leave it. But if you can not do anything, but you can always do your own work along with that, you do it. That's it. So spark is the next one. The spark is your identification about your own self. So identify the spark in you that can motivate yourself. It is from a simple activity like any activity, what you want to. Some student was telling, I'll sleep, I'll see the TV, or the thing. That can. These are all very, very. As I told you, based on the level of understanding, you have this simple alternative measures: watching a TV, going for outing or party, or even drinking tea, all those things. But beyond that, I told you some people may go for outing, etc., like some space. Now, in the COVID period, you cannot go, no? You cannot go even out to the street, step out of the house, tourist places. Of course, we are now a little bit relaxed, but telling that. So you can have some, think about the intrinsic features. Got it? Intrinsic figure. Anyway, all the time, even if you are in stressful condition, even if you are in the lockdown condition, if you are in the office cabin, even if you are in the college, your own body is with you, yourself is with you. You have intrinsic factors to manipulate. You have the powers to manipulate yourself, not the others. That you should remember. You have to manipulate yourself. That is your right. You cannot manipulate yourself. I mean, you cannot manipulate others. That is not your right. That's a hidden agenda here. You cannot manipulate others. So that is one yes under this 3S list. Final is the spirituality. Spirituality is, you don't please take in that a different aspect. I think now people to understand. While uttering the word spirituality, I'm not telling be Christian, be Muslim, be Hindu. That is a what is a kindergarten class, or the middle class, or a higher secondary class, or the now moment is very, very, very crucially involved in deciding you. So I wish all you to take at least this particular last word. You can always think about or not think about other things which we have been speaking so far. I thought this 3S will be able to help you. The last yes is still more important. If you don't like that word spirituality, you can just add this type of, this word there, the now moment. The now moment is all in your control, and you can have experience. Experience even the now moment. You people are all, some people are stressed by these 2 days works, etc. Some people have other stressful events in their mind, all those things. But the now moment, everybody are linked together to this StreamYard, isn't it? You and me are speaking towards each other. I mean, looking at each other or whatever it is, hearing at each other. The now moment is so beautiful. It is so beautiful that you are simply listening or simply speaking. No matter whether anything, I'm talking about the girl saying, if you are able to think about any other thing in your home or mother or any other relatives, you just even come back and be in this moment with a very small attention or awareness. That sort of continuous array of the now moments can also bring changes in your life, provided you complain about these stressful situations or the stressful life that is led by you or undergone by you, like this. So I think, like, as a chapter coordinator and I'm joining hands with your college in bringing about this topic, and you have listened to very eminent speakers who had made their contributions widely in their fields, but for your understanding, they have made their findings in a simpler forms. That because I told my target audience or UGPG or first initial level of research. So their slides are all simplified, and yeah, we have made something before you. We have served something before you. The now moment is like that, and if it is possible for you to acknowledge this, you can take this and you can enjoy this. Again, once again. This is how we are going to live, if at all some ideas from this inputs or the note from me is swallowed up in your mind. So anyway, whatever it is, whichever I told you, you have to have your intrinsic factors, you motivate yourself, this and that. Whatever way you want to bring about it, you just keep it simple. Simple and that is again subjective, but keep it simple. Whatever way you want to be, that simpler way you can take it and be ever happy. So the simpler way, because sometimes we are getting discouraged by certain things like uh, certain things like you have to be no pain, no gain, and this is very hard. You need not be discouraged by that. That's what you me simple decision, stress free. I cannot even say the stress free is life. Stress should be there, isn't it? That's how we need to understand. Anyway, whatever measures you need to take on your own the way, be make it simple and enjoy life. So with that, again, I wholeheartedly appreciate this joint venture. I made by this, because our own department and the chapter and Ethiraj College tremendously they have put all their efforts. Dr. Sujata, secretaries Malati and Sudha, uh, all other members whom the names I do not know, but I have been wondering how eminently they have been working behind the screen, off the screen, on screen, everything. I am really astonished to see their cooperative activity under the headship of Dr. Sujata. It is possible only under, if they are showing their working culture beautiful. I am very much happy. Again, I am very much thankful to our all our secretaries, presidents, HOD, former HOD, and above all the students who are all participating in this to make the venture very happy, and at least some take-home messages there. We will be happy. So with that, I'm trying to conclude this, and online platform is over for whomsoever responsible for the event. Thank you so much.
Thank you so much, ma'am, for sparing your valuable time with us and gracing the occasion with your incredible final note. Thank you once again, ma'am. I now request Dr. J. Priya, Assistant Professor, Department of Biochemistry, Etharaj College for Women, to proceed with the prize distribution.
Good evening, all. Before I announce the winners, I appreciate the effort of all the participants who have come a long way and actively participated in all the offline activities. So, going to the winners of the event, the prize winners of bio cartooning. First place goes to R. Fatima Nasrin, University of Madras. Second place goes to S. Kritika, University of Madras. And third place goes to R. Kartika for Kavita, Etharaj College for Women. For creative writing, the winners are, the first place goes to Kritika S., University of Madras. Second place goes to Glory, University of Madras. And third place to Lokeshwari, Etharaj College for Women. The winners of quiz, first place goes to Amrita Shri and Amrita Lakshmi of Etharaj College for Women. And second place goes to Sindhuja and Amrita Varshini of University of Madras. And third place to Kritika S. and Soundarya of University of Madras. Congratulations to all the winners. Vote of thanks. Are they able to see that? Congratulations to all the winners. It's an honor to have been asked to offer the vote of thanks on the occasion of two-day regional symposium on basic insights of neuroscience against neurological stress. On behalf of the Department of Biochemistry, I thank the management, Madam Principal, Dr. Kodai, and Madam Vice Principals, Dr. D.B. Usha Rani, aided, and Dr. Usha Priya, self-supporting, for their constant support and encouragement for the conduction of the event. I express my gratitude to our co-convener, Dr. Ilangovan Vellaichami, Professor, Head, Department of Biochemistry, University of Madras, for all his extended support and motivation. My sincere thanks to Dr. A.J. Vanishri, Professor, University of Madras, Guindy Campus, and regional coordinator of SNCI Chennai chapter, who served as a bridge connecting Ethiraj College and SNCI, without whom this symposium would have not been possible. I thank Dr. Sujata, Head Associate Professor, Department of Biochemistry, and Dean of Academic Self-Supporting Ethiraj College for Women, who have always been a pillar of support in all our department activities. I thank all the guest speakers for sharing their expertise and knowledge in spite of their busy schedule. I thank all the participants for their active participation in various activities. I place a special thanks to Ms. Kavita for all the technical support rendered for the smooth conduction of the symposium live by StreamYard. I take this opportunity to acknowledge the efforts of organizing committee members who have fulfilled their duties quite well. I once again thank each and everyone who have been the strength of the symposium. Thank you for being with us. Thank you all.
It's now time to listen to our college song. The participants are requested to fill the feedback form to get their e-certificates. The feedback form link has been given in the chat box. Please click the link and get your e-certificates. Congratulations to all the participants and special kudos to the prize winners. Thank you one and all for making this regional virtual symposium, NEUGLIA 21, a grand success.
Ready.
Share. Until next time, this is Department of Biochemistry, Etraj College for Women, signing off. Thank you one and all.